workshop so our colleagues from China could understand what I'm going to say. So first I would like to welcome our collaborators and colleagues from China. I will introduce briefly them for coming to our institute. I will just mention that, well, there are two very important institutions from China. The Institute of Plant Protection from the Chinese Academy of Agricultural Science, and also the College of Life Science and Technology of Huazhong Agricultural University in Wuhan. So we will have Professor ZHANG and Dr. Seju Wang from the Institute of Plant Protection, and from the College of Life Science from Wuhan, we will have Professor Ming and Professor Mei Liu, who will share their work. Besides that, we will also invited two Mexican colleagues from who have been working in the biological control of agricultural pests for many years and have made very important contributions. From Investiad Irapuato, Jorge Ibarra and Cristina del Rincón. She will participate by Zoom because she couldn't join us. And Benito PEREYRA from the University, from la Universidad Autónoma de Nuevo León, from the University of Nuevo León. And finally, Isabel Gomez and Sabino Pacheco, which have been collaborating with us, with Alejandra and myself for many years, and who have now settled their own research lines, and they will share some work that they have been doing in recent years. So with IPP, and we have been, and also with Huazhong Agricultural University, Alejandra and myself, we have a long-lasting collaboration for more than 12 years. And we have, in the case of IPP, actually this year, we signed an agreement between our institution, the Institute of Biotechnology and the Institute of Plant Protection. And we were appointed since 2019 as adjunct professors. So we have a very important and long-lasting collaboration with both institutions. So just to finish, I would like to say that, well, China relied in science and technology to solve problems and invested an important amount of money over the years, which has led to an impressive growth in all areas of knowledge, including agriculture. Recently, China opened international scientific collaborations with different programs. Thus, China has become a great opportunity for establishing important scientific collaborations. We believe that this workshop will be important to show that it is important to seek for an agreement between the governments of China and Mexico to establish a cooperation agreement in science and technology, allowing more scientists to benefit from both countries. So we hope this workshop will be interesting for all of you, and well, I welcome everybody. So muchas gracias, and I will leave now the word to our director, Dr. Laura Palomares, which will be in charge of initiating the workshop.
Thank you, Mario. Thank you. Welcome to the Institute of Biotechnology, your Chinese colleagues, and of course our Mexican colleagues. Thank you for coming and being here today. So it is an honor to welcome you to this institute for this China-Mexico workshop on biotechnology in insects and nematode control. First of all, I would like to acknowledge Professors SOBERON and Bravo for their leadership that has resulted in this collaboration with China, which we see as especially important, not only for the institute, but for the country. This event brings together leading scientists from both countries to address agricultural challenges that affect crops of great importance, including maize, legumes, and vegetables. And I believe that through the exchange of knowledge and collaboration, we aim to develop innovative, sustainable strategies for pest and nematode management approaches that protect both the environment and the human health. I thank all the speakers that will be here today, and I wish you a very continued collaboration and a very success meeting. So with this, I declare this event open, and I give the floor to Professor Soberon to introduce this first speaker.
Okay, so I would like to welcome Professor ZHANG from the Institute of Plant Protection. The talk is Development and Application of Biological Pesticide Products in China. Sí. No tenemos micrófono.
Is that need to open? Okay.
you can change the.
This one,
I think so.
forward is reverse.
No,
Reverse?
no. I thought you should go with this, and this is just a pointer.
Okay,
There's a pointer. Okay. With this,
okay.
so you can change it.
Thank you, Meryl. Thank you, Ali. I'm very, very happy to be here because many years ago we began to collaborate. I think during the last 12 years, we collaborate together, have a very nice publication and young scientist training. The first one is Zeyu, study here, co-supervisor with Ali and Mario. Now he's a young scientist in the country. Another one is Xuebai, last year joined this laboratory. I think it's our honor to be here. We hoped the last year, just like Mario mentioned, last year we signed agreement. This collaboration is enter a new page. We hope in the future both the scientists, especially for the young scientists, join together, work on the biopesticide and a lot of things. So this is where this time we visit here. In the plans, our plans 5 years ago, we planned to visit here, but unfortunately, the COVID-19 stopped the whole world. So a little bit late, but not so late. Yeah. So today, I will introduce some work about development of application biopesticides in China. Yeah, my lab's the same, working on the BT, the Bacillus thuringiensis. So because not everybody working on the BT, so I give some brief introduction about that in China. In China, agriculture is very, very important. So in the field, the insect pests and the plant disease also is very, very serious. Yearly, over the 1,000 different pests occurring in the field. Special, some very serious, over 100 kinds in the fields. And the loss from the pests is great, over 140 million tons in China. So including a lot of the plant disease, which one? Disease, insects, and within recent years, weeds is very serious and some rodents. In the national level, we make sure 19 the top insects and disease. 10 of them are insects, including the 4th armyworm, their hometown from the American continent. 5 years ago, they went to China. And others, for the main crop, especially for the food crop, rice, wheat, maize in China is the main food crops. So have different insects and disease. So the national level, we launched this 19th species. So it means the government pay a lot of attention and give big money to control this and research these insects. In the last almost 30 years, in the whole country, yearly the occurring of the pests over the 5 million hectares. But the controlled area, prevention area is over that because some field need to control twice or triple times. So this is all the situation lasted 30 years. And for the invasion species also is very serious. From the 1990s, the whole country began to open to this world. So become accompany the economic exchange. So a lot of insect, pests, and plant disease moved to China. So this is the situation, the red different color. From this is from the Middle Asia, from the East, North Asia, is from the Southeast, the country. Yeah, different the insects and disease spread to China. So totally, last 3 years, central government support the funding to investigate all the invasive species over the last 30 years. China now over 600 species, including pests, insect pests, plant disease, and weeds. Yeah, over 600. So each year, 1 or 2 species invade to China. So this is the very famous the insects, as you know. So besides this one, insects, after the damage by the insects, so they have some wood. So the fungi invade and on the corn, on the crops. So these crops have no price for the human being and animal foods. Yeah, during the last 30 years in the whole country, we have a Ministry of Agriculture, and beyond that, in the province and the cities and the county and the towns, the 5 levels government have a plant protection system. It's a from the central government to the local government. So for the control, the firstly is monitoring. Yeah. So according to the different stage, from the early warning, agricultural control, biological control, chemical control, ecological regulations, lots. So for the whole country, the monitoring and forecasting system is quite good. Until now, it's established over 1,000 nationwide monitoring regional station located in the different province. And each province send the data to the Ministry of Agriculture to guide the farmer how to use the chemical pesticide and biological pesticide, and to regulate how to push to planting to cropping the different crops, because we can change the rotation the crops to keep away from the disease damage. So this is in the field, the smaller station monitoring and combined with the forecasting the weather or the insect pests and the plant disease. This system is promoting the professionals from the prevention and control. So supported by, so each year central government spend a lot of money to support the different level to do this work. So this system works well. Until now, for the agricultural production, this system, especially for the pest control, this system is quite work well. So this for the disaster special, just like the rice, the maize, and the wheat, they have a different system. So this system is by the professional team. This team from the company, both from the company or from the government. Yeah, this is special for the national, we call the national, the same the control. And so innovation for the method. So in the whole country, about 218 national grain prevention and control demonstration areas located in the main food cropping area, including the Northeast and Yellow River and the Yangtze River. Yeah, another for the ecological engineering system also is very important. They use the biodiversity to control different pests, including the insects and the disease. Yeah, just like in the greenhouse, use the different plant to, this is the tomato. Normally in the greenhouse, the whitefly is very serious, so they plant in the different, the bank plants to attract natural enemy and to repel the insects. Yeah, and I will introduce more about this biopesticide, because the pesticide, including the virus, fungi, and the bacteria, just like BT from its sites. From the 2015, the Chinese central government to push the action, decrease chemical pesticide used in the whole country. They push the different level, including research projects and action in the production. It means to decrease chemical pesticide and chemical fertilizer. So to compare the 10 years change. So for the ingredients and the products of a biopesticide, they all increase the double. Yeah, this is the column is a different. It's 10 years ago, 2014. It's 2024, almost the double. So the policy from the central government to push this and encouraging biopesticide research and production. And for the example, in the last 7 years, different years, registration of new products, both the chemical and biopesticide. We can look at the data. So over the 40% products from the biopesticide. So this is a new situation from the government. And for the biopesticide, normally we know there's from the microbial, from the biochemical and botanic, that this is a different the total products number. This is a gradient from the different the strain and components. Yeah, this is the total products. So this picture shows it's a great improvement of the biopesticides. Registration from this year, end of this year, so March. So for the antibiotics in China, antibiotics belong to the biopesticides. So totally about 2,800 about the different antibiotics. They are function to the plant disease and insect pests to go together. This is the registration. It's a number one biopesticide in China. Yeah, for the registration, the government to increase the cost for the chemical pesticide. Nowadays, for the new products, about more than $200 the cost for the one chemical pesticide. But for the just like BT and the fungi and the virus, about the 20, how to say that? 2 million RMB is $200,000. It's much less than the chemical pesticide. So this use the cost to regulate the registration of chemical, to limit the chemical pesticide development. So this is the compare with the USA and China. This is for the registration, the fee is zero. Therefore, especially for the biopesticide, and this is from the EPA's data. So this is the different the trait compared with the different columns from the registration. So it means the government give a lot of support for the biopesticide, from the research to the production and application in the field. They have decreased the cost. If a farmer buy the biopesticide, government will give some support, decrease the price. It means a farmer only paid less than the normal price. Okay, so for the BT research, I would these products from our laboratory. This we code this number is GO338. These first ones. Genetically engineered BT strain. We transfer the correct 3 agent into a natural strain. Natural strain control the pests as well. So after the transgenic, so they can control both Leptospira and Coleoptera pests. So these products received some permission from the government. Yeah, I just mentioned 5 years ago, the armyworm invade to China, especially for the maize. Is the number one insect pest nowadays in China. How to control them? Because the larvae of these insects almost in the whole leaf, So if we use the traditional spray, the liquid method, it's difficult to reach the whole leaf. But we change the mind. How about the granule? So we use the granule. Maybe it's quite good. Another one, now maize is a number one crop in China because the area is a number one. It's about 45 million hectare in China. The area is a number one. It's much more than the rice. Before 10 years ago, rice is the number one crops in China, but nowadays maize. So for the loss from the armyworms, we cannot stand this. So how about this we need a new method. So we choose to collaborate with the chemicals, pesticide scientists together. They have a good technique to make tiny the granule. So granule now, because this is so big the area, we have to use the drone. So nowadays, in the end of last year, in the whole country, the drone is over 260,000 in the whole country. So now the almost 40% area for every crops used by the drone to control, to spray the chemical pesticide, biopesticide, and fertilizer. So efficacy is very high. So the company from both sides, this almost from the big company, they just make money from the farmer according to a control efficacy. If less than 70, no money. Yeah, the farmer pay nothing for them. It means they need a good efficacy result for the farmer to make sure there's harvest. So this is why we choose this one. So this is the red color. It's easy for investigate. So the diameter is about from the 0.1 to 0.5 millimeter, very tiny. So for each gram, about over 10,000 particle. It's very tiny. The core is a sand. It's a sand. It's very cheap. It's no pollution to the environment. So compare with the wettable powders. Normally, for each hectare, use over 200 liters water. It's difficult to use the drone, so it's why we choose that one. For the cost, it's less than the WP, the products. Yeah, so make the sand is a graded. It's big, so it cannot drift like the liquid, the droplet. So this in the field is working well, and it's very cheaper. So the cost is less than the WP, the products. Okay, for the special, when the maize growing is the whole stage, so it's easy. We can find this the surface and the whole of the maize. So if some the drone and some the rain left is easy to dissolve. And in the partial times, the concentration is much higher than liquid sprayed. So special, this is for the egg, it can stick some the particle. So when dissolved, so the larvae, the neonate, when they hatched, they have no future. Yeah, this is the surface of the maize. Yeah, so surprised results show us because the BT products can kill the Spodobacterials less than the third instar. But in the field, we can easily find the 4-star, 5th-star insect can be killed easy because we conclude the concentration is much higher than liquid method. So it's easy to control, that's one. So how about the trait of this granule? We use it in the Yunnan area because its altitude is very high. Sea level is above 2,000 meters, similar here. After a week, we can compare. This is 3 days, this 7 days result. Zheyu did this work last year. So after the 7 days, for the WP products activity, because the BT proteins degrade by the sunlight, by the UV light, it's much. And the activity lost than 50%, but granule keeps 70 more. So this is another trait. So it means the toxicity of granule is much higher than WP the products. And in the Inauguration by the people, we call the artificial sprays, use the bottle, we spray the small hole, just like that, like to push the pepper, the cooking times. Yeah. So the result is much good than use this granule. This is the WP white balls powder results. Okay, so this new formulation now is registered. Maybe the end of this year we can get the formal certificate. So compare with the traditional formulation. So BT the tiny granules by the drones can decrease the pesticide, can save labor. Efficacy is quite good, and these products can avoid degradation by the UV light. So these products also can use other insect pest controls now. Okay, for the further prospect now in the national levels, a lot of big companies, just like Syngenta and a lot of big companies, they transfer to conserve the biopesticide. They have different products now developing in the different level. So for the world levels, so in the future, the experts project over the big market. So from our side, we think that biopesticide research and production should increase the demand for the organic food, strict management for the chemicals, pesticide, and enhance the environmental awareness and the requirement for the sustainable agriculture development. Another is improvement of quality and efficacy for the products. And the BT is quite good in the biopesticide products, but others have sometimes limited in the different conditionals. So efficacy is not so good. Yeah. So we should pay a lot of attention about to improve some trait of that. So what biopesticide our farmer need? So we concern in the country. What need? They concern both the food crop and the cash crops. Yeah. The quality of biopesticide is the stability persistence and ingredients. They hope to spray when the times can kill the insect and control the different insect and the different disease. So they only save the labor and save the money. Yeah. Evaluation of the controlled efficacy for this biopesticide, especially for the new one, we should pay a lot of attention about that. Not like BT over the 80 years application in the whole world. So another one is should pay attention about the application technology. Yeah, because compared with the chemical pesticide, they can use any time. Yeah, for any stage of the insect, but the biopesticide has limited. So when, how to use, and when to use, how to use, and it's very, very important. So this is a concerned technique. We should pay a lot of attention. So this is a picture in the field. So in China, a lot of farmers have kids. When the kids younger times, they spend their times in the field. So the father, I guess they have two kids. The smaller one is with the mom. Maybe they cannot walk. The big one is with the father. So special for we know the biopesticide is beneficial for the human being health. Special, I think, firstly for the kids. So their childhood in the field. So decrease the chemical pesticide and give a nice condition in the field. I think from the childhood is very important. So this is our responsibility. Okay, thank you very much. Thank you.
No sé si hay alguna pregunta. Some questions.
What about, can you comment about the price of the tiny granules compared with the chemical insecticides or something? Some comparisons with the price.
Okay, it's a tiny granules. A tiny granule, the price, I think it's a little bit less than the WP, the products. Yes, the cost for the farmer, for the production, is no increase. Yeah, it's good. Yeah, but the efficacy is high. Normally we can decrease some the usage in the field, so it also we can decrease the cost for the farmer. Yeah. Okay.
Thank you very much for your talk. When you refer to pest, it's only insects, not phytopathogens? Are you working with phytopathogens?
Yeah, I'm working on the BT because the BT is limited, only can control the insects. For the pathogen, not in our lab. Yeah.
But in your institute, it is working on the control of phytopathogens?
In the control. Sure. Oh, yeah, in our institute, we have for biopesticide, two hands. One hand, several scientists, several laboratory control for the insect. Another for the pathogen, plant pathogen. Yeah, you have just like Bacillus subtilis and some fungi from the Huazhong mildew. Yeah, different fungi products against to control the plant disease. Yeah, but not from my lab. Thank you.
Thank you.
Thank you.
Yes, over here in the front.
Yeah. Okay.
Yes, so thank you for a wonderful talk.
Thank you.
So I understand, and it is very interesting. I have, excuse me, I have never seen before the use of these pellets instead of the liquid formulation. So I wonder, of course, they're more stable and that makes them more efficient, but the insects must eat them. So do they kind of like them? Is there anything there to make them appealing for the insect to eat them? Because I see that they accumulate in these particular parts of the plant. So instead of being spread over the leaves, where as you showed in your pictures, is where the larvae likes to go and eat.
Yeah, special for the this granule, tiny granule, special for the fall armyworm, because the fall armyworm after the hatch, their neonate move to the whole leaf. Yeah, they stay there because they are we call knockout. This kind of the insects normally is active in the night. So they're scared the sunlight or something. They are only in the hole of the plant. Yeah. So if we can spread the granule to fall down to hold the part, yeah, because in the field, so in the midnight, some the 露水叫什么？忘了，浊。露水是 no, yeah, it's just the rainfall, the water, or some the humidity can make them dissolve. Yeah, so if they just like the small part, yeah, on the leaf, yeah, if they eat it, so they have no future. Yeah. Normally, the BT kill insects need 2 or 3 days, but for this granule, 1 or 2 days can kill, no matter how it's from the neonate to the 4 star or 5th star. Yeah, this is according to this insect the action. Yeah, we made this granule formulation. Thank you.
Well, thank you for the presentation. It was very interesting. And I want to ask you, because I thought here in Mexico, I think, as I've known, biopesticides are not very used, really, or at least not agriculturally or economically, globally. Maybe there are some tests, but mostly in universities. And what do you think will be the incentives to improve this technology, and what are the difficulties to improve it, like economically and technologically talking?
You mean this granules?
I mean, in general, the biopesticides, because here is mostly used the biochemical pesticides, and I mean that. Sorry.
Okay, it's a very nice question. Nowadays, for the BT products production in China, these products fermentation and industries commercialization, the cost is very cheap. This products for the farmers can compared with the chemical pesticide. Just like each hectares chemical pesticide need 100 RMB, but the BT also can reach this price. Not like other products that are much higher than chemical pesticide. So this is why BT products is the #1 the products used in China as a biopesticide. So especially, yeah, we consider this application two-hand. One is for the food crop, another is for the catch crop. You mean if the farmer who to make the big money from the field. So the catch crop is very important. And another, nowadays in the greenhouse, special for the vegetable, for the catch crop, is developed very, very, very nice the level. In the whole country, about how much? About 5 million hectare area, a greenhouse in the whole country. So in this kind of the greenhouse to produce the expensive products, so farmer like to use the biopesticide because you can recover big money from their products. Yeah. So others, just like fungi, insect pesticide, the price still high, but not like a BT. Yeah, BT is real cheaper compared with chemical pesticides. No problem. Yeah.
Okay, thank you, Jie. I think we should move forward with the next talk. Thank you very much.
Thank you. Thank you all.
So the next talk is Benito Pereyra. So we have time for more questions. There are like several questions left. So during the coffee break. ¿Esa es la tuya, verdad? During the coffee break, you can ask him, okay? Thank you, Benito.
Thank you, Ale. We'd like to thank to Dr. Alejandra Bravo and Dr. Mario Soberon for the opportunity to stay with you this morning. I was in this institute for many years ago, 33 years ago, many years. Okay. Well, my presentation is evolution of insecticidal crystal proteins and parasporins. Okay. Well, the world is a colossal laboratory where many organisms, maybe all organisms that live in this planet has DNA as material genetic, inclusive pumas, especially tigers, BT and myself. This picture was taken last night. Okay, but concern the several questions about BT is what could be the reason of the wide specificity and very wide lethality of this bacterium against several organisms. This is more or less the BT toxin spectrum of action. Start with Lepidopteran, after Dipteran, after Coleopteran, after many others ones, and lastly, in special human cancer cells. Okay, the first questions was awarded with a simple question: what could be the reason of the wide spectrum, and what could be the real reason of the many shapes of the crystals and many kinds of proteins? But all these proteins kill cells, insect or human. Okay, then a search of the genes reported, we found that the BT synthesizes many kinds of the proteins with the biological activity, okay, and lastly, parasporins, okay. A total of 1,000 of proteins with some kind of activity. In this sense, we select a special kinds because there are some reports in the literature where especially Rudemacht and others report that for cry1 maybe exchange of especially domains. Okay, as you can remember, the crytoxin, the typical crytoxin, is composed by three domains. One for alpha helix, two beta sheets, and the three, where was demonstrated that the domain 2 is involved in the specificity especially, and the domain 1 is responsible for the pore formation in the membrane. After, the ALEJANDRA teams reported that maybe true that for toxin type CRY1, the different toxin is resulted from different combination of domains, especially domains. Okay, but however, we are very interested to know if this event could be shared with another toxins. Okay, in this sense, I like very much this. If you want a better answer, ask better questions. No? This is absolutely correct, no? in one sense. No? Okay. Okay. We use a different BT strain with different crystal shape, toxicity, specificity, and antibiotic resistance spectrum. So we have the strain A and the strain B with different crystal shape, toxicity, and specificity. Okay, we need many microbiological experiments to come to this. Okay. After that, we select according its resistance and show the crystal and toxicity. Okay, some of our results are that. In green is the donor and the crystal shape, and several another ones are recipient, and this is the results. Okay, as you can see, HD 225 is crystal shape is irregular and was mixed with GM7, bipyramidal crystal, and this is some results. Okay, in some ones, the crystal was modified, the shape. This is some results. As you can see, this is very different shapes of the crystal. This is squares, and this is the original crystal of GM33 and GM43. Bipyramidal typical, and with a molecular mass of 130 kilodaltons. The results of bioassays against Trichoplusian E show that this is the original strains, and this is the transconjugants. Okay, the more spectacular result was obtained with HD73 as receptor and donor GM33. Okay, as you can see, GM33 has not activity against Trichoplusian E, but however, we have another result that could be interesting like that. Yes, that when HD225 displaying some toxicity, okay, not at 100% like typical donor. Okay, the results show us that the shape of the crystals and the toxicity in the nature may be the results of the intensive genetic flux among the strains, and this flux may be done in the nature and the insects. Okay, and some another of the results show us that not all strains can be used as donor, neither as recipient. Okay? It's necessary a lot of work previous, but it's important that all of the strains tested just this could be used as donor. And interesting, HD225 could be used all for recipe. Okay, this is part of the reason, because in our institute we have a big collection of the IBT strains collected from different places of the world, especially from agricultural fields in Mexico. Okay, another thing, and lastly, in our lab we're working with special proteins called parasporins. The parasporins are crystal proteins had not activity against insect, at least to those who are tested. Okay, but common Lepidopteran, 2 or 3 Lepidopteran, and 3 Dipteran. Okay, then according previous papers, especially those of the Japanese teams, were described for first time as parasporins. Okay, there are especially 2 kinds of parasporins. In figure A, show the typical protein composed by tridomains. This is good for PS1, PS3, and PS6. And another kind, like aerolysine, beta pores. This is the typical shape, domain 1, domain 2, and domain 3. Domain 1 is involved in the specificity, domain 3, and another domains resemble the cry one. Okay. This is maybe involved in the first time the pore forming. Okay, in our collection, we found four strains that maybe synthesized some kind of parasporins. The first 4 was purchased from the Dr. Mizuki from Japanese collections, and these strains, and the Japanese was tested against several cellular lines. We are very interested in two leukemias, mold 4 and your cat. Excuse me. Okay, the IB84 and IB88, all collection was tested with specific primers in order to detect several PS, and this is the results. This one, an IB79, was not recognized for any. However, this has biological activity. Okay, this is the main results. This is the first 4 are the holotype strains obtained from the Dr. Mizuki lab, and this is the results with our strains. This is very important because in black is the line cell line HS27. This is normal human cell line, and this is for malignant melanoma and JORGE. Okay, these results show us that our strains has a good activity against these cancer cells. In this sense, we tested with Apotox Glow, a triple-X from Promega. And with this kit, we can evaluate viability, necrosis, and apoptosis. In this sense, the results show us that our strains, according viability and necrosis, has a very good activity, but especially activity IB84, yes, and IB88. Okay, after that, we performed the kinetics of the diet. We choose the strain AB84 for the next experiments. Okay, and the next experiments, we performed microarray assays in order to detect the expression of the genes in cells treated with the toxin. Okay, this is the sum of results. Yes. And very important results are involved with several signals, and less for involved in metabolism, especially in signal. And we select several genes, genes whose RNA expression was higher, more than 10 times in comparison with the control. Okay, and after that, these genes were tested with qPCR in order to demonstrate that the results of the microarrays is true. Okay, after that, compare the results of qPCR with another chips, and with the results, we formulate an putative mode of action of PS4, and we found, according with the results, the first time the toxin requires a binding to some receptor on the surface. After oligomerization, more similar for toxin CRY1, after signal cascades, especially those involved with signal for calcium and other insulin signals, and especially syneases. Okay, this was a negative regulation, but another has a positive regulation, especially response to epoxia endocytosis, taurine synthesis, and ubiquitin-mediated proteolysis. Also, has an impact on cell proliferation. In conclusion, the putative mode of action of these strains fit the lysis model by losses of membrane integrity, while GM88 is involved in apoptosis mechanism. Biological activity of PS4 and from our strain IB84 on leukemia cells, triggers biological process related with damage caused by beta pore forming toxins. where the increase in the intracellular calcium appears to be a key element for activation of different cellular strains. Okay. We like to thank with Astrid Espino and Ezequiel Lozano, two excellent students, CONACYT and PAICYT, and obviously Dr. Eiichi Mizuki and Dr. Chiro Kumura from the Biotechnology of Food Research Institute, Fukuoka Industry. Okay, that's all, and say.
Time for 2 questions.
Thank you, Benito. So I didn't follow well. So parasporin is able to bind to this putative receptor in the plasma membrane.
Yes.
Is there a possibility that it binds to the membrane itself, or it has to be through the receptors?
Yes, it's a good question, but I believe that these are receptors because this protein has not any effect on another cells.
Yeah. Okay, so have you looked to the, thinking about all these kind of responses that you can have in these cell lines, have you looked, you said that it must involve probably calcium changes and this hypoxic response. Have you looked at the calcium changes in response to this treatment?
Yes, the most probably is that.
Yeah, thank you.
Here, thank you for your talk. I'm just curious about the assay in which you are combining 2 different proteins. I mean, what does it mean to be a donor and recipe? And I wonder if this shift in the crystal shape is related to, the protein are mixed between them, or the folding of the protein change, or what does it mean? I'm curious this.
It's possible that both kinds, because typically crystal shapes is formed almost exclusively of one kind of toxin, similar Cry1. As you remember, Cry1 is formed for the typical 3 domains and 130 kilodaltons. The crystal shapes, I don't know if for all the strains, but those studied for us are so. These strains, and you can see, the pyramidal has a protein of 133 kilodaltons. Although these proteins has not activity against Lepidopteran.
Okay, and just why is the shape important? I mean, it's important for the function.
Okay, mainly function. Yes.
Okay, muchas gracias, Benito.
Okay, thank you very much.
So the next speaker is Dr. Min Soon. A ver, yo le pongo. Okay, thank you.
Okay, thank you. Good morning. It's very happy for me to come here to visit Mexico and this nice institute. And also thanks Dr. Ali Bravo and Dr. Maria SOBERON, our old friends, to host this workshop, and so let me have this good opportunity to come here. Then I give us report something. What would have done in bacillus thuringiensis for kids as a resource? How to use this resource to develop biopesticide, or especially nematicides. For the title, the proteins with nematode activities is early given me. So I will expand this, expand it to a discovery of a novel BT and develop a new nematicide. We know that BT can produce a very good, crystal, and this crystal can be used to kill insects and nematodes and some others like cancers. So for these characters, so BT was developed as the largest microbial biopesticide. We can see so used to control insect, and also what I right now talking as to control nematodes. And so its crystal protein genes was successfully used for in transgenic crop to control insects. For BT, this species, we can say we have reported more than 71 serotype and 84 serovarieties, and 3 kinds of insecticide was developed used to control Lepidoptera, Diptera, and Colapted insects. But no report before my talk or something time stone. No report to have developed a nematicide to control nematode in plant protections. But in my mind, we think the toxin diversity is quite strong or power. So the too many kinds of active ingredient exist in BT species. So here shows the contribution of Chinese to develop to form the new serotype. There are totally 11 or 12 serotypes was founded in China. In my group, I work as Professor Yu Ziniu is my supervisor. Right now he is retired. He has developed 3 kind of new serotype. So for the crystal protein genes, you can say for the published, we can they have more than 80 groups was released in the as a holotype crystal protein genes. In the internet of the PPRC, they have also showed some or described some nematode proteins. You can see in this list. I just downloaded it. You can see most of we know that nematode crystal protein is like the Cry5, Cry6, Cry14, and also some others like Cry8 or Cry21. So in this case, even the no nematode was commercial, but the crystal protein genes was we have people have found about at least 30 years ago. But we can say that nematode is actually, especially in China, the plant parasite nematode disease is the second largest crop disease in China. Maybe the first, maybe the fungi. And for this kind of disease, we can say nematode infect the root of nematode. So this is very not easy to find that, and also it's very difficult to control. So it causes a huge loss for annual years. And we can say this shows some lifestyle for the nematode, or say plant placid nematode. We can say for and the left is a larvae. In the second larvae, it was introduced to the root of the plant. When they colonize in the root, they'll be developed as the adult, and then to infect the growth of the plant. So in this case, one it's invaded to the root, so it's very difficult to control the nematodes. So for currently, for the existing products to control nematode, most of us use the chemicals. For those of the chemicals, most of them is very toxic. So according to this, in China, China's government made some government policy want to use a green development. So want to control, make decrease the use of high toxic chemicals, and it encourage to develop bio and nematicide. In this case, there are right now in China there have many laboratories is doing try to develop bio nematicide and they encourage them to use it as a commercial product. So in this case, in some ways, when we doing so many years work on BT, so sometimes we also ask a question: how large will be the diversity of BT in nature? So in this case, if we want to answer this question, we must obtain as we can a BT large amount of BT strains. So in this case, we try to find in some way exhaust the effort to isolate the BT strains from the soils as we can. So in my laboratory, we have a long history, especially from my supervisor, Professor Yu. We have a long history, more than 40 years, to collect BT strains. Right now, we have collected more than 20 or 40 soil samples from all of China. In some way, also when we give a report, especially when we face the students, we encouraged them to scan this code, and if they scan that, I will tell them how to collect soil samples, how to ship the samples to us. If the students send us 5 samples, soil samples, we will charge 100 renminbi to his handphones. Yeah. So after we collect the soil samples, who can do that? How to use the soil to isolate the BT? So in my laboratory, we open a training course. This training course mainly based on those the new students, for example, the junior under, or the first time come to my laboratory. For those students, the first courses is to isolate BD strains from source samples. Currently, we have at least 315, near to 400 students have taken this training course. So in this case, we can get a large amount of BD strains. Based on this training course, they can do something like isolation, identification, genome sequence, and cloning expression, the crystal protein genes. Usually for these courses, it will take 3 months. So, and also for these training courses, or let it become teaching courses. And here, after the student takes these courses, we are giving a certification. It may be a benefit for him to find a job. And also, we also use this kind of courses to develop as virtual stimulation courses. We also awarded as a Hubei province virtual stimulated courses. So we have 5 models for students can learn in that as a virtual study. So this shows the face of the virtual simulated courses. So in this case, we can get a huge amount of BD strains. 2 years ago, we have got 20,000 BD strains. Right now, we have 50,000 BD isolates in our hand. So in this case, if I compare it, also compare with the public data, I can say I have the most BD sources in my hand around the world. So if we can do that, when we get the BD strains, so select some of doing genome sequence. After we get the BD strains, well, identification whether this one is different from other one. We use the 30 molecular markers to identify whether this one is different from the other one. Right now, we finished the genome sequence about 10,000 BD strains. So in this case, we can find too many of new type of crystal protein genes. This data shows 3 years ago. We say for the public the data, there are 80 groups of crystal protein genes. But in my hand, we found that at least more than 200 groups of crystal protein genes. And this data is growing every week, because in my laboratory the training course is always going on. When we're doing this kind of work, we also find some rule how the crystal protein exists in one strain, and what kind of the crystal protein co-occurrence. So in this case, we found some genes sometimes exist together, especially for those genes have a same spectrums. So in this case, we can in some way we can predict those haven't done activity, so can maybe predict its spectrum or activity. So in this case, we have so much bit strings, and also too many of the crystal protein genes. So we can we develop some mining systems to extract or predict the crystal protein genes in a string after genome sequence, and also predict what is the activity or spectrum for the string or for the crystal protein genes. So in this case, I'm honored to take part of the nomenclature committee. So along with Dr. Bravo. So in this case, because we have so many kind of crystal protein genes, so in the nomenclature we can provide some the detailed information. So if when we get this so large BT string samples, and also we can get big data. So we can use these two information or strings to drive development of new microbial pesticide, especially for those nematode. Here I give two samples. The first is developed a new nematode of BT-HAN055. Firstly, we doing by manual the biopsies to isolate or screen the high toxic strain to nematode. After that, we can use the bioinformatics to predict which one shows high toxic to nematode. For in this case, we got in some way high toxic strains, HAN055 in about 10 years ago. When we get that, we have set up a long pipeline. So first we got a string as a library, and I use some. We developed 4 kinds of analyze tools like BT barcode, BT toxin scanner, BT toxin digger, and toxin sorter to predict which one is with the best or high toxic strains. In this case, we got these top strains. We named HAN055. When we doing when we select these strains, we are also doing something like mechanisms. Why this strain has high toxicity to nematodes? So in this case, in my library, we found actually many kinds of toxic crystal protein genes or like virulence factors or some small molecules. They also shows the toxicity to nematode. And also we studied on the mechanism, how what kind of pathway what kind of receptor for this the toxin to target nematodes. So after that, we're doing some field trade to use the control nematodes. We also developed a foam powder to do that. So in this case, after 80 years hard work, so this strains was registered as a nematocide in China. This is the first one registered as only targeted to nematocide in China. Maybe also say around the world. So in this, for this kind of work, we expand the commercial BT products from control insect to move to nematode. So it's lucky for us. In the last year, we got the Hubei province first prize for technology invention. So this kind of recommended by Chinese government to use to control nematode in vegetable and soybeans. This is the second. This is the first case for us to develop a new kind of nematicide. The second is a kind of small molecular. It come from all around in many kinds of natures. These chemicals named tranexamic acid. This acid we found is very high concentrated in corn and sugarcane, but for other crop near to zero. So when we found this kind of chemicals in BT strains, we found this is high toxic to nematode. So right now we have found the synthesized gene cluster, and also we transfer this the gene cluster to rice. And there's also found this the rice also resistant to nematode. We also at the same time we also found this. We named this TAA. It can inhibit the hatching of nematode egg, and also can award the root node to if we use this to treat the seeds, can award the root node attractions. So this means this is what we have done. When this gene cluster when transferred to rice, it can show resistance to root node nematode. So right now we are doing this work for registration. I think this kind of chemicals, this is a small molecular. Actually, the structure of molecular weight is very similar to citric acid. And for this kind of molecules in China, we register as a new kind of chemical pesticide. We use an echinacin. So if we register it, maybe they have a new kind of nematicide, is echinacin. So for all the registration biopsies or test experiment, we passed half of them. When we're doing those kind of like toxic environment response and some other safety test, these chemicals also is very very safe. So we have expect it can be get the registration in the end of next year. So both of these cases showing us we have been and continue to use to strive to discover more BT strain to meet the needs of pest control. So in this case, when we can develop a new pesticide to control nematodes. So in this case, we also ask another question. Besides BT, how about the other spore-forming bacteria can be used as a biopesticide? So we can see for those spore-forming bacteria in all around those bacteria kingdoms, we can say they have more than 100 genus, more than 60 species exist. they can forming spores. So next we will try to use a big sample or big data to develop more kinds of pesticide. Right now, in Chinese government encourage us to do that, and also give a large money for us to do this kind of work. Last year, Chinese government support us to about $66 million to build an agricultural microbial resource bank. We will build a new building. This new building will be finished at the end of next year, or also as 72 months later. So maybe for Ellie, if you come to our university in 2027, you will make a new large building. So this case it shows in our university, we form the advantage of agricultural microbial cluster. We have like this agricultural microbial banks, and also we have a national key laboratory, a national engineering research center, and also inspection or testing center for microbial products, and also some major scientific and technology instrument for agricultural microbiology, and also some others. So in our university, we have a large group of research or teachers or students to work on agricultural microbials. So in our university, we also faced the two lines of agricultural production. The first is the crop production. The second is breeding industrial. For all those the working line, they also most of them faced like a disease, animal disease or a plant disease, and also nutrition supply, and the environmental remediation or productions, also food quality and safety. So in this case, previously or mainly currently used to chemicals to deal with this problem. In our university, we try to use the microbial to overcome that kind of problem. This may be used greatly for us for the green development and to meet our happy life. So this is in Chinese. I don't know how to do it, how to translate it. It means use the first we have research resource, and then doing research, and then to development, and they make them together. Okay, thank you very much. This is our group. We have in our group we have 3 professors. We have more than 70 students, so it's a large group. Okay, thank you.
Okay, thank you. We have time for questions.
Thank you very much. Very, very impressive what you've done. It's a very good idea to use or to be helped by the students the way you did. I have a question about the strain, because sedentary nematodes don't eat bacteria, do they?
Actually, for plant nematode, they can't eat bacteria.
Which one, sorry?
For plant nematode, nematode, they can't because of
they
still
can't,
the stylet.
Exactly. They
looks
don't
like
eat bacteria.
very small. But in my mind, in the soil environment, the one BT used to around the soils, they can secrete some small molecule, and also they can infect the microbiome for the reservoir. We have done that kind of work. They can reprogram the metabolic of the soil bone, and also the root node, and also the plant. They also to fit each other, to use to adjust the environment of the soil, and to use this to control the nematode.
Okay, thank you. And about the TAA molecule, that is a soluble molecule that the bacteria produce, yes?
Yes.
And that's what has the effect on the eggs being soluble?
Yeah.
And is it produced in enough quantities in the soil? Have you tested that?
What?
On the soil? Because you tested it in the lab, yeah?
Yeah.
But in the soil?
BT, we found some BT can produce this kind of chemicals. It is molecular.
Yeah.
Yeah. And one way, if the string produces more TAA, so the activity for user control in the pot test has a high activity.
Fantastic. Thank you very much.
Okay.
Another question? If not, I have one. Because, well, you know, the main problems with BT toxins is they're using transgenic plants is the evolution of resistance. So your collection has a great value for searching genes who can skip resistance or who can counter resistance. I wonder if you are doing anything, or have you thought about mining for special Lepidopteran or Coleopteran pests that are very important for transgenic plants to search for new toxins from your collections to skip resistance, for instance?
Thank you. Actually, we have done that, but for the biopsy is a more huge work than I to isolate or identification for these genes. So sometimes I want to encourage the government to support this kind of work, but the government say you can do it by yourself. So I want to try to encourage some company. But currently, for those large companies, they don't care that. They said, if you found someone, I can use too much money to buy that. But before that, we don't do that. But also, we found some currently in China, they have some company. They're doing some work like to transformation for plant. They're very interesting for our crystal protein genes.
Okay. Well, thank you very much. Congratulations.
Thank you.
No sé. So the next talk, I think, is Jorge Ibarra, no? This is before the coffee break. No? After his talk, we will have a coffee break, and we will continue.
Okay, gracias. Well, first of all, you can hear me? Yeah? No. First of all, I would like to thank both Alejandra and Mario. It's always a pleasure to be here on the IBT in Cuernavaca, and also because I've known, besides Alejandra and Mario, some other people here, very, very important. I like to talk about, well, after seeing Dr. Ming-Sung's presentations with the overwhelming amount of data. Mine will be almost a case study, okay? So I'm going to show you also my main research line is mostly on insects. But we have been working with nematodes in the last, 6, 7 years. And we found some important, I think, important data that you might be interested on. So, well, should I change it here? Yeah, okay. So as Dr. Sun already mentioned, that there were until 1976, all the BT known were always active against Lepidoptera. In 1987, there was discovery of BT israelensis with the mosquito activity, and then in 1992, Dr. Krieg in Germany discovered the Nematode strain of activity against Coleoptera. After that, there was a lot of discoveries. Some of them were very interesting because there were reports and even some patented strains against the ants, cockroaches, slugs, rattlemints, and nematodes too. Mostly because of this company that was called Mycochin in San Diego, California. And they weren't very well supported with data. So we kind of believe or not believe that they were really active against all those range of different insects and other type of invertebrates. But nowadays, we can now be sure that, for example, our subject, nematodes, are very well supported because mostly Dr. Zhang recent review, which is wonderful, was published this year, what shows a lot of important results, and this review is wonderful. I would recommend you to check it. The question that somebody over there, I don't know who, asked about how to select the nematocidal strains of BT if the, this works? Yeah. Because this is the structure that the plant pathogenic nematodes feed on. It looks like a straw. It's called stylet. And it's something that is very difficult for them to eat bacteria. They eat mostly inside each cell that they get into. So it's quite difficult to isolate BT that are toxic to nematodes because of these nematodes do not eat bacteria. So when we are interested in selecting BT strains with nematocidal activity, we have to use definitely this one. It's Rhoditis elegans, C. elegans, which is a model, and it eats bacteria. Okay? That's the first step. So according to the list that Dr. Sun has in the review, these are the main nematocidal proteins that are already well characterized, and these are the most important ones, and he showed that in his presentation. Cry 14, Cry 5, Grind 21. Those are the most important ones. And the problem is how to use this nematode or plant parasitic nematodes to see if they are actually active, because they are not only active against this Synrarity, but also with some animal parasitic nematodes. Okay. So this is more or less what we see when BT active against nematodes looks like when tested against C. elegans. This is normal one. This is the kind of thing that we see when they are active against the nematode. And of course, most of them are killed. In our case, we have a much more modest collection of BTs. It's a little bit more than 1,300 strains in our collection in Simvestab. And we practically selected 310 strains, practically at random, except that none of these strains produce the beta exotoxin. Beta exotoxin is the one that is secreted in during the vegetative growth. And that toxin is very active against nematodes, okay? We have tested that, and even we used that as a biological form to select the strains that produce the beta exotoxin. Beta exotoxin is not allowed to be used in the field. Well, at least here in Mexico and in the United States. In other parts of the world, they are, but not here. So after these 310 strains we tested, these 10 strains showed some activity against nematodes, this nematode, C. elegans, okay? But these two were the ones that showed activity within 90% and 100% mortality. So we selected these two. It was our first selection. And we showed here the percentage of nematodes in each biological qualitative bioassay, okay? It could be seen that they have a good activity, good nemacidal activity. When we added 100 micrograms per milliliter of the spore crystal complex, okay? This is our control with one strain that we know that is not active against the nematodes. Maybe you could wondering why there are more than 100% nematodes in some of them, because this the BT, when it is not nematocidal, the nematodes grows on BT, okay? That's something that good news when we see something like this, okay? Okay, once we separated or selected those two strains, first of all, we checked what was the LC50, the lethal concentration at 50% from the purified crystals of the LBIT-596 was 24 micrograms per milliliter, okay? And we, in a very preliminary screen of the cry genes that had that strain, we found the cry 5B, very well known as a nematocidal, with a 99% identity of most of the cry 5B known to date, okay? So it looks like there was nothing new about that. But this one, the LB107, had an activity more or less the same of the later one. It's an LC50 of 25 micrograms per milliliter. And this preliminary scrutiny of the cry genes, we found this cry14A like. It was like because it was only with 90% identity. Okay? And for cry14, it is already well known as a nematicidal, but it has some difference with the ones that they were already known. So taking into account that we decided to keep working only with LBIT-107, okay? Why taking that decision? Well, in a previous report, sorry about this picture, but it was taken from the article. This is a result that they found. This is the group of Rafael Royan. These roots of tomato were transgenic tomato transformed with the cry5b, and it was good, yes, but there were still some galls. This is Meloidogyne nematode, which is, by the way, the most important nematode in the whole world, the plant pathogenic nematode. There was some galls here. See, this is the control. This is the transgenic tomato. And there were some few, but they were still there. So they didn't show the normal transgenic BT toxin when we compared with the insects. Okay, in insects, we have this one, a transgenic plant with BT, and there is no insects at all. Yes, or maybe 96, 97% control. So at this moment, the transgenic plants are the more effective ways to control insect pests. Here, it wasn't that big. So we decided to keep working with the other one, with the other strain, which showed this kind of effect against this nematode, which is C. elegans again. Okay, we see all the effect that it has in the intestine. This is the control. Okay, so we decided the morphology of this strain was kind of atypical, not very well usual to this morphology. The crystal was attached to the spore, okay, and we started to sequence the genome of that strain. Only one, not the 8,000 that Dr. Son did. Okay, and we found some interesting results here, even surprising, because we found that it has the cry21, which is also known as a nematicidal. Of course, the cry14 that we already had discovered, site 1A, chitinase. Chitinase is also, they might have some activity against nematodes, as I will show later, because we didn't know actually when many years ago, we checked this bacterium, same strain with chitinase activity, and was the highest one between all the different that we checked. And that might be important. We don't know yet. And more important is this BMP1, a metalloproteinase, which also has some activity against nematodes. Okay? So it looks like there were some kind of bacterium that has a lot of different weapons against the nematodes. So we cloned separately the cry14 in criterion 1, and we found some spore and crystal complex, two different when they were together, as the one I showed you before. And we tested the activity separately. This is the cry one. In this case, there were the typical bipyramidal crystals, the cry 21, and we still didn't make the bioassays against with the cry 21, and it shows some activity. Okay, this is the control. We show some activity with the cry 21 that we have already cloned. So what about the SID1A? This is the control, and I'm sorry about the quality of this picture again, but the only thing I can say is that mortality was very low, but it didn't grow at all. In this case, with E. coli, it grows very, very well. But with the SID1A, the nematodes were there, but just stopped growing. Okay, so they have some activity. When we tested in combination, it looks like when the combination of BMP1 and CRY21 shows some kind of synergism, still not as good as the one with the whole strain. But still, there was some combination that it looks like possible synergism, okay? According to some results before that we have published, where we found that the BT can be translocated into the plant if we added the spore crystal complex in the root of the plant, okay? And now we are working also in that one. So we decided to use the whole strain with tomato plants, and where we added for 20 weeks into the rhizosphere of the plant, okay? 20 weeks, another one during the 10 weeks, and the control. So, was it translocated actually? Could it, we found here in the foliage? Yes, we were able to obtain the strain into the foliage. So it was actually translocated. When we check the plant, once the plant was grown, the roots, this 10 weeks is control, this one with the strain 107, it looks kind of different. It looks apparently different, much better than development of the root than the control. This is at 20 weeks, okay? If we checked the galls that were shown, I'm sorry, this is Meloidogyne again, I didn't say that. These are the galls of Meloidogyne, okay? You find it here. And apparently there were more in the control than in the ones subjected to the LBIT-107. Of course, it was necessary to make a quantification of that number of galls, and we found something, well, it looks interesting. With the 20 weeks of inoculation, the number of galls decreased significantly here. And with the 10 weeks, again, not as much as with the 20. And the control, of course, there were much more. were more or less 170 galls. This is an average, of course, per root. So it looks like this strain has by itself a good nemacidal control of the most important nematode in agriculture that are in the world. At this moment, we have a construct where there's a vector for the protein expression in plant, okay, to make a transgenic plant, where we included the CRY21 plus the BMP1 protein, okay? And we are at this point, we need some funding to keep working on that. At this moment, Mexico is kind of a critical part. But this is maybe next step, because we believe that this kind of problem with nematodes in plants can only be controlled with transgenic plants because of the way they eat, as I showed you, or using something like the approach I showed you by making an endophytic bacterium getting into the plant tissues. Okay, and I think I break the score. I mean the spectrums, because that's it.
Preguntas. Mira, vaya voy.
Hi, thank you very much for your talk. I'm curious about these endophytic BT. Are they normally endophytic, or is this a particular strain that you found that colonizes the plant?
Yeah, that's a very good question, because I'm working on that. The thing is that this particular strain was not found as an endophytic bacterium. But in the last 4 years, we've been working with endophytic BT strains.
So like you kind of evolved them to become endophytic or something like that?
It looks like it is something like that, because, for example, just to give you an example, from 110 wild plants in the field, we found 22 strains that were endophytic. That was their natural habitat. And there is BT. And also, at this moment, we are using those endophytic bacterium, which, by the way, most of them, they don't have any activity, any insecticidal activity, no nemacidal activity. But they have the, let's say, the machinery to express the CRY proteins. And that's what we did in the last 2 years. We introduced a CRY, which is toxic against Lepidopterans, and in an endophytic strain, and it worked beautifully. It was expressed, formed the crystal, and what is more important, more interesting, is that once inside the plant, it can be transmitted vertically from the seed to the next generation, I guess, via the spore.
That's very nice. We have population of Meloidogyne enterolobi here in the lab. If you would like to test that, we can talk about that later. But
oh yes,
yeah.
Yes, I will be very interested.
That's a very one of the bad emerging pathogens, nematodes.
Of course.
Thank you. That was a very nice talk. So just taking again the same questions. So this is endophytic, but it's mainly localized in the plant root, or it can be endophytic in any other parts of the root of the plant?
In the whole plant.
In the whole plant.
Because we obtained the plates that I showed you from the leaves.
Yeah.
Yes,
Yeah.
and the inoculation was in the root rhizosphere.
Yeah. Okay. So another short question is, I'm curious about the chitinase. So the chitinase is secreted. What could be the role in controlling the nematodes?
The thing is that nematodes have a lot of chitin, not only in the what it could be the exoskeleton. It is called actually exoskeleton, but it's not like the arthropods. But the whole exoskeleton of the nematodes is made out of chitin. And there are some other parts of the nematode with chitin, both in the intestine and some of the reproductive apparatus of for copulation, for example, they are made of chitin. So we didn't make those genes cloned to test them, but maybe that's why this strain works much better when there is the whole strain and not all the different genes separately. Okay?
Okay, we don't have more time, sorry, because we are extremely late. So we have 20 minutes now, from now. We are extremely late, no? So we have to speed up. So 20 minutes break, because we have 6 posters that you want to see, at least a few of them, if you can or not. And we will come back in 20 minutes, okay? So, and then in the second part, I will ask the speakers to be faster if they can. Sorry. Okay. I think we can start, Cristina.
Okay, are you ready?
Ajá. Because, so I want to keep the time. Eso hay que voltearlo para acá, ¿no? No, porque es muy bueno para el speaker. Ajá, sí. Ah, es que es Cristina ahí. Ahorita es que, ¿sabes por qué está Cristina? No, déjalo así, pues como estaba. Sí, es que te vemos en la pantalla, Cris. Ajá, tú no nos estás viendo, pero te vemos en la pantalla. Ok, entonces...
¿Apago mi pantalla?
No, está bien ahí.
Ajá, okay.
¿O prefieren que la apague y que vean...? No, está bien, está bien. Okay. O sea, ahí te vemos a ti en las pantallas chicas y en la pantalla grande tenemos la presentación y tú la vas a ir moviendo, ¿ok?
Perfecto, sí, claro que sí.
Ok, entonces vamos a empezar nada más para mantener el tiempo más o menos. Faltan de regresar, pero ya les dijimos, ahí vienen. Es que como había pósters, están todavía un poco discutiendo, ¿no? They are still with some posters, but they will come while you were talking, okay? Mario, please. So, bueno, quiero introducir a Cristina y aquí está su plática, ¿no? Ok, del CIMASTAB Irapuato, sí. No, es de Guanajuato, Universidad de Guanajuato.
Ok, I can initiate.
Please, go ahead.
Okay. Firstly, I want to say thank you to Alejandra and Mario for this invitation to participate in these workshops. In this occasion, I want to talk about my research lines for 35 years, firstly in CIMASTAB Irapuato and after the last 16 years in the University of Guanajuato. We are working one special type of virus, specifically a virus infected invertebrates, mostly insects. Exist 4 families important with potential bioinsecticides, poxviridae, rhioviridae, iridovirus, and baculoviruses. All invertebrate virus, like another virus, have material genetics. Maybe in some occasion is DNA, and another occasion is RNA. And all of them is inside and protein capsids. Most of them are virions, and this is a special characteristic of the virus pathogenesis to insect because it's very large viruses with genomes of 100 and 200 kilobases. This is special because it's very different from the virus to affect the humans, for example. And this virus have another particularity, have an occlusion body. This occlusion body protects the virions for the environmental condition of the another environmental effects. One family have the more potential to develop in this activity with bioinsecticides, the member of the baculovirus family. These viruses have DNA double strand, circular supercoiled genomes, and I said in the last moment about 80 to 180 kilobases. The virion is a rod-shaped nucleocapsid with this size, and the polyhedra is possible visualized in the optic microscope because have a big size, more or 1 micron. Exists for the moment 4 genera of this virus recognized for the ECTV committee. The alpha baculovirus, this genus is affecting to the Lepidoptera species of insects, especially pests of the great importance in the agriculture systems. The beta baculovirus are another type of virus. This virus is more small and exists 2 more genera, the gamma baculovirus and delta baculoviruses, specific to Hymenoptera and Diptera insects. This is the forms, and you can visualize it, the forms of the virions. This MPV is called multiplers or single SMPV. Why? Because exists 1 or more virions in size and enveloped and very different amounts of virions, and maybe 100 in some cases, into in one occlusion body. In contrast, strains are defined but having a single nucleotide. This is an example, including only one virion for envelope, but different virions inside of the occlusion body. The granulovirus include only one virus for occlusion body, and the form of those occlusion body is different. Exists these forms is the polyhedra forms is typical to the virus infecting Lepidopteran species, but we isolated another different strains with the occlusion body have a different form to the polyhedra, maybe these triangular forms. Occlusion body is very important for baculoviruses because are highly stable and can resist more normal environmental conditions, thereby allowing virions to remain infectious indefinitely. Even was isolating for some researchers virus with activity for 40 years. The granuloviruses are capsules more small when compared with the polyhedra from occlusion body, because this is only 0.2 or 0.4 microns. Evidence suggests that occlusion bodies can survive at passage throughout the gastrointestinal tract of bears. We can facilitate their dispersion. The occlusion bodies consist of a special protein and very high expression of these proteins in the very light process of infection on baculoviruses. And it's possible this protein constitutes the 40% of the total expression of the proteins codified for this type of virus. How this virus infect an insect? Only for injection of the foods contaminated. Ingestion of occluded virus for the larvae is the first step. Inside of the mid-gut, the high pH, the proteasase enzymes, and another factors dissolving the protein of the occlusion bodies, calling polyhedron for MPV, and granulin for the granuloviruses. These virions dissolves infecting the columnar cells in the midgut on the insect, and the first infection occur in these tissues. The virions progeny is called gem virions. These virions, via hemolymph of the insect, infect in a second round of infection the rest of the tissue of the insect, forming a new occludium virions, and the larvae death for the global infection occasionated for this type of infection. This is the typical aspect you can observe when the larvae is infected with baculoviruses. It's typical. The larvae cling to the higher parts of tree, including exposed place they will normally evolve due to the rest of predator. But this is an advantage for the viruses because this behavior provoke the larvae drip down onto leaf, which will be consumed by new hosts. This is a special behavior, and in this moment we know this behavior is induced for genes codified for the baculoviruses. How is the production of baculovirus? Exists two different mechanisms using insect cell lines, but it's very expensive, this expression system, and the lower production cost is obtained with the in vivo production. Why? Because standardized production technology is established in this moment. Exists different insect colony to produce this virus in artificial diet. Production of alleged variety of virus exists. No establishment of new cellular lines is required, and many baculoviruses produced in vivo have found commercial success in different agricultural systems. This is some very fast panoramas or scheme. The baculoviruses or commercial production of baculovirus has been carried out at this moment only in vivo. In vivo, I comment, is very expensive. Either the first step is apply the virus in a specific diets, semi-synthetic diets or natural diets, depending of the pest, in the laboratory, and this diet contaminated is incubated, depending on the level of infectivity of each strain, for 5 or 7 days after harvesting of viruses occur, and the formulation quality control is necessary to evaluate using bioassays to determine what is the level of virus we need to apply in the fields, in different fields. I want to emphasize in my work, I'm in this moment is the more important line is working to baculoviruses isolated of Hospedoptera frugiperda, the fall armyworm. Why? Because the corn production in Mexico reached 23 or 24 million of metric tons. The 60% of this production comes from a small scale production, and this producer needs options less expensive when compared with the use of the chemical insecticides. Mexico occupies the 7th place in the world in the production corns, and specifically in here in Mexico, the main states produce this grain are Sinaloa, Guanajuato, Estado de Mexico, representing the 61% of the national production. I working in the University of Guanajuato. In these states, Guanajuato ranks 5th in the entire context in the production corns. And unfortunately, this grain corns is attacked for the different places, but the fears of the more important pests to attack, to affect the production level of corns, not only in Mexico, if in all world, is the fall armyworm. These pests, unfortunately, the chemical insecticides have been used, but obviously they caused resistance in the exons because farmers usually perform an average of 6 applications of these synthetic insecticides during the growing seasons. Obviously, we have many problems about with the abuse of the use of chemical insecticide. This is the cycle of Hospedoptera frugiperda. The cycle completes dependent of the temperature of the year station. The average is 25 days. So, what are doing about this line of investigation? For approximately 15 years, we are working with a collection of strains, some of them exotic strains from Argentina, from Honduras, from United States, and native strain collected for my laboratory and the person who working with me in fields, in soils, in larvae, and in different samples obtained for the producers of corns. The collection in this moment have 7 strains with different level of virulence. This is the name of the strain. We compare the biological activity, but 6 initially, but the last year we are isolated a very high level of virulence, a new strain, maybe a new strain. We continue characterization this strain and recovery from the soil. The difference were in the LC50 were characterized by restriction fragment parents initially. Obviously, was necessary to the whole genome sequences to compare these strains. The objective of our investigation is to establish the level of infection of this SFMPV baculovirus strain. First, in the cell lines, insect cell lines S9, perform phylogenetic analysis with 3 conserved genes from these strains, determine the LC50 of this strain, analyze in vivo and in vitro proteomic profile derived from SFMPV baculovirus infection, and finally, in this moment, we are working in the sequences of the whole genome of these strains. The larvae we are rated in a semi-synthetic diet with German basket. The insect is maintained in these incubators, and with respect to the phylogenetic relationship, with the first approximation for my lab was using 3 very conserved genes from the all baculovirus sequences at this moment, the left 8, left 9, polyhedra genes. Using this conditional PCR, were established what is the relationship with this different geographic difference origin of this isolated. The first step consisted in the DNA analyzed with different restriction enzyme. Why? Because this technique is very fast, it's nothing expensive, and we obtain good and precise information if the strain was different or not. And this is a good approximation. The second objective was established the LC50, and I want to destack this exists at this moment the 2 strains, the SFMPV isolated from Argentina, and the last isolated strain, the SFMPV, here in the Guanajuato state, because the LC50 is very, very low. Only 1 or 0.2 occlusion virus for a millimeter squares. We are tested this virus in rapid tests in fields in a small area in some community here in Guanajuato, and the field tries efficiency of these strains reach level to 80%. When we compare this level effect with, for example, spinoteram and chemical insecticides, no exists significant difference at statistical levels with the viruses and the chemical insecticides. This is the good news for us. The permissibility of the cell lines was analyzed too, and you can observe all the strains infecting these cell lines. It's possible to observe the occlusion body inside of the nucleus of the different strains in these cell lines. But we continue to establish what is the relationship with one strain of baculoviruses infect Spodoptera frugiperda larvae to select what is the more virulent, the more effective strain to be developed in formulations, pesticides to apply, bioinsecticides, to apply in the fields of corns. Double dimension protein gel were made to select proteins present in infected insects and compare them with not infected insects. Obviously, the spot markers in blue were analyzed for spectrophotometry to analyze and obtain the sequences of these proteins. This is only some examples for the extraction for RNA and the cDNA for making macroassays. These macroassays proportion us very valuable information because we detected the differential gene expression, and these differential genes, some of them are negatively regulated by the infection, but another genes have regulated by the infection. What type or what class of this gene in baculoviruses is a regulator product of the infection of viruses. Genes imply in apoptosis regulators, cytoskeleton regulators, sickle size, RNAi transport, and reactive oxygen species. In more detail, we detected a specific gene I can explain in the next slide to not consume more time. The first approximation was obtained with the SDS page gels. Obviously, it's in one dimension only, but we observe with respect in the line 2, one baculoviruses of strengths. Reference strain is SMPV baculoviruses. And our perfils proteins of Spotria frugiperda, the occlusion bodies, was completely different when compared with SMPV viruses. This is good news for us for the identity of this strain. This is another type of gels. But after we make the two-dimensional genes to analyze the proteins at different times post-infection, 12, 24, and 48 hours per infection. This is a gel shows what is the protein expresses without infection at 12 hours. Why this is important? Because this protein is involved in the correct metabolism of the insect, and maybe this protein is turning off for the infection virus and impossibility the insect to defense of this infection. For example, these proteins, protein disulfidosumerase, vesicle associated membrane proteins, this protein is important because it reduces the oxidative stress of the larvae. And interestingly, when compared with the protein and the same time post-infection in isolating from larvae infected with this strain of baculoviruses, some of these proteins disappear and another proteins appear, especially myosin heavy chain protein, enoyl CoA hydroxylase, and peptidyl propyl cistrines protein. Why this is important? Because this protein is not being described in insect. Obviously, is codified for the viruses. This protein affect, for example, degradation of fatty acids in mitochondria. This loss related with overproduction of mitotic membrane permeability, and this facilitate the introduction or penetration of baculovirus into the different type of tissue cells. For example, peptidyl prolyl cistrine isomerase overexpression in the HeLa cells cause lysis of cell. Maybe this is the same function of this protein in the tissues of insect infected with this virus. At 24 hours post-infection, without infection in the healthy labs, we detected this type of proteins. All of these proteins are very important in the correct metabolisms of the insect. For example, related to vesicle triumphant, overexpression in the late stage to control damage for pathogens or for other organisms. But when this compared with the larvae infected with the baculovirus strains, for example, this protein, muscle-specific proteins, is related to the recognition of actin and the cytoskeletal formation. See, this is important because we connected what happened, what the virus introduced in the cell, and what happened not only with the gene, even with the expression of the protein for codified for this gene. All of this information was published the last year for our lab, and this is another example of this protein. What was the results about the phylogenetic comparison between the strains? In the first approximation, using only the 3 conserved genes in all baculoviruses, we obtain this phylogenetic reconstruction. All of this strain is very similar, see, as ubicated in the same cloud but in different subclouds. If the difference, even if the difference is only 2 or 3% of the whole genome, in this 3% of difference, we found different activities, maybe in relationship with the level of difference present into this strain, because the level of difference is completely different. Okay? When continued with this work, we establish in this moment, we obtain the whole genome of 6 of these strains, and this is only the first results to the annotations of the genomes, but it's interesting because we confirm using only the 2 conserved genes the same result when we are using the whole genomes. The percentage of identity is at the level to 98% in average. Well, I need to speak more fast because I want to only very fast to show it what is the results in the first approximation in fields. We prove only 2 strains in this moment. One is the Mexican strain and the Argentinian strain. This is the effect observed in the fields when we apply this concentration of occlusion virus per hectare and compare with the effect of spinoteram insecticides in fields here in Mexico. Interesting for us is when you compare what is the effect of the formulation of baculoviruses, we prove at this moment 2 formulations, liquid formulation and emulsificant formulation. The liquid formulation is the more infectant in our field tests, and if you can observe, maybe at 5 days, the effect is not good for us for an strain when compared with spinoteram. But at 7 days post-application do not exist a significant difference in the results about to the mortality of the larvae when we apply the formulation of baculovirus and the chemical insecticide. This is very good news for us because proof these strains is very usable or at least have a great potential to use in applications in the fields. The last line of investigation in my lab is developed for one PhD student. This is only preliminary results. We decide to make a combination of the more virulent strains of baculoviruses combined with the more Bacillus thuringiensis strains proportioned by Jorge Ibarra at Investiad and prove different proportion. This is the isolated Argentino mixed or combined with the ABT-13 strain. In this proportion, the LC50 expected in this combination was times lower with respect to the LC50 observed. The LC50 observed was 8 times lower with the LC50 of the combination. And this is an evidence and maybe exists and synergisms between the baculovirus strain and the BT strain. Why? Because when estimated the LC50 of both pathogens but in separate way, the LC50 is more higher when compared with this combination. With the other strain, Sinaloa isolated, this is a Mexican strain, similar results about to relationship with synergisms was observed. In the 10 Bacunium graphic, you can observe the synergism is located in the area of this effect. This is a promise for us to continue with this investigation line. especially focused in Spodoptera frugiperda due to the importance of this pest. But in my lab, we are working with diamondback Plutella hilostella to control this pest in cruciferous plants, especially broccoli. And the conclusion at this moment is the SFMPV virus strains are highly similar at DNA level, but have very different levels of virulence. Maybe we need to investigate in which consists this difference of level of virulence. We studied in this moment maybe 10 different genes implied in the virulence, especially PIF genes, EGT genes, BHF genes, and another protease and chitinase in the occlusion virus present in this strain. Maybe 2 or 3% is very low difference, but it's possible, and this low difference exists different genes implied in this virulence. So it is important to first select the most suitable virus strains before carry out more application at the field. So they are effective in controlling Spodoptera frugiperda, since not all of them have the same potential. Well, we observe this situation in the lab in first instance. Finally, I want to say thank you to my group of investigation in agriculture and food biotechnology lab here in the University of Guanajuato. In this moment, I have two postdoctoral positions, Dr. Angeles and Dr. Jonathan, and the three of these girls is my PhD student, all of them working in the same lines, one of them in molecular biology specifically, and another in the field 10s in the fields of corns. I work in a stretch collaboration with Dr. Elisabeth Ernieu in the University of Tours in France, because she helped us to the sequences of the genome, and for 1 year I practiced my sabbatical year with her in France. Okay, thank you so much. Do you have any question? It's great for me.
Thank you, Cristina. We have only time for one question.
Okay.
No questions?
No questions? Okay, then we will continue with the next speaker.
Okay.
Thank you, Cristina.
Thank you.
A ver, entonces voltea la pantalla para acá. So our next speaker is Dr. Mei Liu. Omar, ¿nos ayudas con el micrófono, por favor? He will put the microphone in here.
Good afternoon, everyone. I'm honored to present my work here. My study mainly focuses on a foodborne pathogen, Listeria monocytogenes. Today, I'd like to talk about LISKNR plays a role in LM growth at cold temperatures. Firstly, let me briefly introduce LM. LM is a gram-positive and rod-shaped bacteria, and is a facultative intracellular bacteria. Interestingly, these bacteria strains are highly flagellate and motile at low temperature, such as 30 degrees and below, but typically at temperature of 37 and above, not motel. Importantly, LM is a foodborne pathogen cause human and animal dysterias, a kind of disease. The symptoms include gastroenteritis, meningitis, encephalitis, septicemia, and abortion in humans and animals, especially some people, including elders and neonate and immunocompromised, such as HIV patient, and the patient received transformed organs. It disease with high cases fatality rates, 20 to 30%. LM is a typical cold-tolerant pathogen that can grow and reproduce at temperature as low as 4 degrees. That brings great risk to food safety and human health. So, exploring the mechanism of LM growth at cold temperatures will provide a theoretical basis for the formulation of measures to control LM growth at cold temperature and the development of low temperature growth inhibitor. Two component systems is a major signal transduction devices in bacteria. One TCS normally composed of a histidine kinase and a response regulator. Typically, the signal triggers HK autophosphorylation at one histidine residue followed by phosphoryl transfer from the phosphohistidine to an ascorbic acid residue in the RR. Our preliminary study and other studies have shown that LASQR, a kind of TCS in LM, plays a role in regulating the growth of LM at cold temperatures, while the special mechanism remains unclear. This study aims to reveal the mechanism of LASQR, this TCS, in regulating the growth of the LM at cold temperature. To conduct this study, we use LM isolated from food start strain named LM201, use as parent strain, and then we constructed 4 strains, including 2 deletion mutant and their respective complementation strains, totally 4 strains, strains 1, strains 2, 3, and 4. And then we carried out the growth curves to all the strains at 4 degrees and 37 degrees respectively. The results show the growth of deletion mutants HK and RR was slower than that of the parent strain. Whereas at 37 degrees, no differences across of strains. To investigate the mechanism of the green diffraction, we performed the RNA sequence for the deletion HK deletion strings and the parent strings grows at 4 degrees. RNA sequence data showed the expression of flagella genes in the HK deletion strings was upregulated compared with the parent at 4 degrees. So we carried out the motility of the swarming cell for the deletion string and the parent string at 4 degrees and 27 degrees respectively. The result showed at 4 degrees, the swarming area diameter of deletion mutants on sulfur agar plates were bigger than those of the parent and the complementary string, where at 37 degrees, no differences across all strings. Furtherly, we observed the flagella by transmission electron microscopies for the HK and RR deletion strings and the parent strings at 4 degrees and 37 degrees. The deletion mutants had more flagella, where the parent and the complementary strings had fewer flagella at 4 degrees. And at 37 degrees, all strings no flagella production. And meanwhile, we measured the transcription levels of the flagella genes of the string at 4 degrees by RT-qPCR. We can see the expression of flagella genes in deletion mutants, either in HK deletion or RR deletion, was higher than that in the parent and the complementary strings at 4 degrees. Above results suggest in HK or RR deletion degrees, the flagella expression could contribute to the growth defect. We know flagella biosynthesis is ATP instinctive. In order to use the energy, in order to conceive the energy, cells may prioritize the initial growth and reproduction in cold stress. So we conclude the TCS in LM, LSKR inhibit the expression of flagella genes to induce flagella reproduction, and the energy is used to promote LM growth and reproduction at cold temperature. Further investigation in the specific molecular regulatory mechanism of the TCS, LISKR are currently underway. That's all. Thank you for your attention.
Thank you. Do you have questions?
Thank you. I have one question. Is it still possible that these two components has an effect on other genes necessary for a fitness of these bacteria at 4 degrees? Not only for flagella biosynthesis. I mean, in the transcriptome, did you find any other target of these two components?
This TCS-LSKR is a, how to say, 多能性的, how to say, multiple function. We, in fact, the RNA sequence data not only show flagella genes upregulated, but also show PTS transportation systems, the genes down regulations. So this just show the flagella genes upregulating the mechanisms.
Okay, thank you.
You're welcome.
Thank you. So as far as I know, this Listeria monocytogenes, it requires the actin polymerization for the movement. So this change in the number of flagella in the bacteria, does it have an effect on the motility of Listeria? The mutants, the LIS-K and ART mutant, do they have a phenotype in the motility? Do they move faster or slower?
Motility aside, we have carried out the deletion mutant moved faster, and the swarming area diameter is bigger than their parent at 4 degrees, but at 37 degrees, no phenometers. No different across all strains, either deletion or the parent strain.
Okay, thank you very much. I think we will move on.
So now we move to the presentation of Sabino Pacheco.
Okay, well, thank you for the presentation, Alejandra. Well, I'm going to talk about some difference in functional difference in crytoxin receptor that we found used in CRISPR-Cas for genome edition in insects, especially in Aedes aegypti, and I want to compare with other results about Lepidoptera insects. Okay. So, all we know that Bacillus thuringiensis is a salt bacteria, and it produces different insecticidal proteins. Some of them are produced during the vegetative growth, and this protein is secreted to the extracellular medium, but also during the sporulation phase produce other proteins that are accumulated in crystals. This bacteria is used for control insects for almost 80 years. And the most abundant toxins, or BT toxins, in the nature are crytoxins. We know more than 700 different genes of crytoxins. Some of them are active against Lepidoptera, Coleoptera, or Diptera, or even for Nematode. Actually, we have 2 talks about this topic in the previous presentation. All these proteins have this structure. It's a three-domain structure. And for this future, it's proposed that these proteins have the same mechanism of action. And this mode of action is proposed that after the ingestion of the crystal, then there is a solubilization to release a protoxin, and these protoxins are activated by proteases. This toxin can bind to different proteins in the membrane, and this interaction triggers the oligomerization to form the prepore, and then this prepore then is inserted into the membrane to form the pore, which causes the cell die. There are several proteins proposed that are receptors for crytoxin, including cadherin, aminopeptidase, alkaline phosphatase, and also some members of this family of protein ABC transporters. In theory, if we block any of these steps in the mode of action, we can generate resistance in insects. However, the most common mechanism of resistance, which is reported, is the blocking the toxin receptor interaction. And I want to present some nice reports about these mechanisms of resistance. The first report of resistance, what this report? This group finds that Eliotic virescens was resistant to cry1AC toxin, and this resistance was linked in a mutation in the cadherin gene by insertion of one retrotransposon in this gene. And it conferred more than 600-fold of resistance. 10 years later, the same group reported that there is an additional mutation, a deletion of 22 nucleotides in the second exon of this ABC transporter, the ABCC2. And this mutation conferred even more resistance in these insects, almost more than 10,000 folds of resistance. So that just suggests that this receptor is more important than cadherin. And another two examples, which are not related to mutation in the genes, but is related with the downregulation of the receptor. For example, this group found that these three strains of Eliotic virescens have downregulation into ALPs, which are receptors for crytoxins. And this downregulation also correlates with low activity of ALP in the brush border membrane. And another example is this one with the cabbage looper, which is resistant 1,000 fold of resistance. And this, in my opinion, is very interesting example because these insects decrease the expression of this gene, APN1, but in order to compensate this loss, increase the expression of APN6. So it was proposed that there is a compensatory effect to mitigate the fitness cost, and thus confer the resistance to the cry1AC toxin in this insect. All these mutations that I mentioned are natural mutations. 10 years ago, some research group started to use CRISPR-Cas to analyze the functional role of the receptors. And I create this table. You can see there are many data, and it took many time for me to make this table, so I will take some time for explaining this table. For example, for a long time, we believed that cadherin was the main receptor in the mode of action of the crytoxin. But with CRISPR-Cas, we realized that the knockout of this receptor in the fall armyworm or the Ostrinia fornicalis doesn't have effect in resistance because they are still sensitive to the toxins. But when we knock out the cadherin in Helicobacter parmigera or in Bombyx mori, we generate resistance. But in the case of Bombyx mori, only we generate resistance to cry1AB, but not for cry1AF. Even more interesting is the knockout of ABCC2, As you remember in the previous slide, I mentioned that one mutation in this receptor confer resistance almost 1,000 folds of resistance. But the knockout in ABCC2 in these two insects only confer resistance 3 times. So there is a huge difference. But that is only for these two insects, because when we generate the knockout in Spodoptera frugiperda, Ostrinia furnicalis, Bombyx mori, or the cabbage looper, we have a higher resistance. So here there is a differential role of this receptor. But it's more interesting, even more interesting, is that if we have a double knockout with the paralogous gene ABCC3, we have a double knockout here, we can increase the resistance even 50,000 of resistance. In the case of Helicobacter parmigera and Plutella gilostella, more than 8,000 fold of resistance. In the case of Spodoptera frugiperda, the knockout of these two receptors didn't change the susceptibility in comparison with the ABCC2. That means that this receptor is the more important for Spodoptera frugiperda, because there is no difference in the double knockout. And the same is happening with Bombyx mori for Cry1A toxin. The same effect we can find with a double knockout of ABCC2 and cadherin. We have even more resistant with a double knockout than a single knockout in the case of Helicobacter parmigera and also with Ostrinia furnicalis. Also, there are proposals that ABCB1 is a receptor. This is very interesting because this protein was identified that are involved in resistance but in Coleoptera insect. But the knockout of this receptor in some Lepidopteran insect also confer resistance. For example, for Plutella gilostella and for Bombyx mori. And in the case of ABCA2, it was reported that some mutation in this protein confer resistance to the Cry2A toxins, and it was confirmed by CRISPR-Cas knockout in Helicobacter parmigera, Pectinophora gossypiella, and Trichopusiani. And also other studios have performed the knockout in ALP or APN and confer resistance, but no in all resistance. For example, in Spodoptera frugiperda, there is no change in susceptibility when they have the knockout in APN. So with this table, we can see that the mode of action is very complex, and for some insects, some receptors are more important than others. But what happened in mosquitoes? I mentioned before that these toxins are 3-domain toxins, so it's proposed that the mode of action is very similar. And actually, the same receptors that were proposed, there are receptors for crytoxin active against Lepidoptera, also are the receptor in Diptera, especially in Aedes aegypti. Actually, there are many reports that propose that cadherin, APN, and ALP are binding protein for mosquitocidal crytoxins. But regarding ABC transporters, there is no information about if this protein is involved in the toxicity. So we want to explore this possibility using CRISPR-Cas to study the functional role of crytoxin in Aedes aegypti. This is the strategy that I used to generate the mutation in Aedes aegypti. First, we collect the eggs of Aedes aegypti and were injected with a complex of Cas9 and single-git RNA to target any of these receptors. After hatching, we call this insect G0 and was outcrossed with a wild type to obtain the generation 1 in order to isolate the mutations. If we detect similar mutation, identical mutation, we encross this insect to obtain the generation 2, and here we have a mixture of heterozygous, homozygous, and wild type. We isolate the homozygous to obtain the generation 3, and then we can study the role of some receptor with this population. The first candidate was cadherin, which, as I mentioned, was we believe that this receptor was the most important in the mode of action of crytoxin. So we obtained 5 different mutations. We select a deletion of 50 nucleotides in the second exon of the gene of cascading. But after several generations, we realized that only females were homozygous mutant, and the male were heterozygous mutant. And that's why, because in Aedes aegypti, the chromosome 1 determines the sex in the insects. And this is because the chromosome 1 contains this locus M, and all the insects that contain this locus M are males. In the case of females, contain this locus M with a show lower letter. So in this case, we mutate this allele from the female, and that's why we can obtain only females homozygous mutant, but no in males. In the case of this, always will be heterozygous. But anyways, we use this population because unless we have homozygous females. But before to analyze the effect in toxicity, we want to analyze the effect in the development of the insects, because it was proposed that it was impossible to obtain one knockout in cat hearing in Aedes aegypti because it's necessary for the development of this insect. So we analyzed different parameters, including oviposition, hatching, development to adults, and if there is a difference in development to males or females. And we don't see any difference. So in conclusion here, the knockout in cat hearing is not necessary for the normal development of Aedes aegypti. So the next step was analyze the sensitivity. We used 4 different cryotoxins, which are mosquitocidal toxins. And we used 2 doses, the LC50 and 2-fold the LC50. But we don't see any difference with these strains. But remember, in this population, only the female are homozygous. So we want to see if the survivors in this experiment was female or males, but we don't see any difference in female or male. That means there is no correlation between toxicity and the sensitivity to cryotoxin using the knockout mosquitoes. Actually, we determined the LC50 for all these toxins, and we don't see any difference between the 4 toxins and the population. So in conclusion, cat hearing is not necessary for the toxicity in Aedes aegypti. So the next receptor was 1-ALP. We choose this protein is MALP. It was proposed that this receptor is a receptor for Cry4B toxin. In this case, we obtained 3 different mutations. We select the deletion of 10 bases, and this mutation correlate with a lower ALP activity in the Bruce border membrane. And we obtained a result similar that had cat hearing knockout, because in any cases, using 4 different cryotoxins, we don't see any difference in toxicity. So the conclusion here was that ALP neither significantly modified the susceptibility to mosquitocidal toxins in the Aedes aegypti species. Another group, they generate the knockout in 2 APNs, and also they observe that the knockout, the single knockout or even double knockout, doesn't change the susceptibility to cryotoxins, specifically to 4BA and 11A toxins. So then, if cat hearing, ALP, and APN are no receptor for this protein, what is the protein that is the receptor for these toxins? So we want to explore if the ABC transporter at the receptor. So first, we identify all the ABC transporters in the genome of Aedes aegypti, And then we perform one phylogenetic analysis to identify the orthologous ABC transporter to those involved in cryotoxin resistance to Lepidoptera insects. So we detect ABC-A2, B1, C2, and this receptor that also is involved in resistance in Lepidoptera insects. But when we analyze the expression in the midgut cells, only we detect the expression of ABC-A2, ABC-B1, and ABC-C2. So we select these 3 different receptors to generate population knockout and analyze their role in the mode of action. But we obtain similar results. We generate the knockout mutations in these 3 proteins, but when we analyze the susceptibility to these 4 cryotoxin, also we don't see any resistance to these to these proteins. So in conclusion, these 3 ABC transporter neither change susceptibility to mosquitocidal cryotoxins. So, in summary, what we know using CRISPR-Cas9 to study the cryotoxin receptors. So we know in Lepidoptera, in many Lepidoptera insects, some receptors are very important. Some of them produce more resistance if we knock out this receptor, for example, ABCC2. But when we obtain the knockout into double knockout, we have even more higher receptor resistance. But in the case of Aedes aegypti, we don't find any resistance linked to the mutation in these classical receptors for cryotoxins. So at this point, we have more questions than answers. It's necessary to generate multiple knockouts similar to Lepidopterans. Maybe there is a redundant role I was proposed for Lepidopterans. Or another option is that maybe other proteins that are not identified are the receptor for this toxin in Aedes aegypti. Or finally, maybe we are facing a different mode of action in mosquitoes. We need to work with these 3 questions. So finally, thank you for listening to me.
Preguntas, questions?
Thanks for your presentation. Very solid results.
Thank you.
I didn't follow you in the ABC, the slides when you showed the ABC knockout but for mosquitoes. There is an expression, no, the previous, yeah. So why, I didn't understand your figures totally because there are some genes expressed higher than the one you choose. So why? I didn't know what's the x-axis suggest.
Here we use RNA-seq data that was performed by another student. And here we detect a higher level of expression in ABCA2. TPM is transcript per million. But ABCB1 is almost in the same level of ABCA2, but ABCC2 have a lower expression.
Okay.
This is RNA-seq data, transcriptome data.
So what is that one, like 5043? It's expressed higher. Beside ABCC2? No, beside ABC, yeah.
This one? This one?
Yeah, that's not ABCC2.
No, ABCC2 is this one.
Okay, that is the number of genes.
Yes, exactly.
Okay, I understand.
And I write the name of the protein here in red letters. And ABC2 have a lower expression in comparison with other proteins, with other genes.
Yeah, it's very interesting. You did lots of mutations, but
there is no link to resistance.
Yeah.
I think, in my opinion, redundancy is very important. And
yes,
that's
we
why,
are working with that.
doing that, you know, the work that is doing, Michelle is doing these double mutations will be very important to test, because I think if you, when you silence ABCC2 and ABCC3, let's see what's happening, no?
Exactly. Maybe we can find a similar mechanism than Lepidoptera and insects.
Or maybe we should silence this, the one that Soju is mentioning. This is in the midroot. In the family of ABCC2, this gene that is highly expressed maybe could be very important, and maybe that is the target.
Yes.
Yeah, thank you.
Okay, thank you, Sabino. So, deja la computadora ahí. So now we move again the computer, and it's Soju 1. ¿Dónde dejó el aparato? Okay, now it's your turn.
Thank you. Okay, so thanks for the organization of Alejandra and Mario's group. It's very happy to come back here again, and also I'm very excited to meet many old friends here. Sabino, Isabel, Blanca, Marigalman, Emiliano, yeah, Jorge, yeah, sorry, yeah, because I spent 1 year here in 2016 to 2017, so I still remember the first day I came here in last time. Emiliano drove me to outside to have lunch and show me everywhere. And also, Mali Galman and Arlene, she's not here. They helped me a lot for that year. Because I'm not good at Spanish, so I cannot go outside by myself, though they always accompany with me. I'm very appreciative for that time. Yeah, so today I'm going to talk about actually, it's 2 parts of my work. The topic is beyond the toxin receptor interactions. The first one is host response, and the second part is commensal bacteria. So what is their act in the BT mediated insecticide action? So I'm showing this picture, but I will not talk about it because Sabino and Isabel, they are experts about this. So I only show this I only showed this picture is to notice that the model of action of cry proteins against the Lepidopteran insect pest has been already finished by the group of Alejandra and Mario. So actually, I'm not sure whether Alejandra still remembers that just before my leaving from here to China, I had a talk with Alejandra, and I asked her what happened after formation, and she told me very few knowledge is known since that at that time. So that's why I started to work with the host defense response from the target pest to the cryotoxin. So after I got my PhD degree, when I started my postdoc, I worked on this part. So last year, so in 2021 there is a good review from Spain, from the group of Guanfele and Patricia. So they published a good review to talk about the defense response. But actually, even they give a very good hypothesis like a model, but actually there are very few evidence of publication to support all these responses. So, since 2019, I started to work about this part, regeneration, because we found a phenomenon to show that after the treatment of cryotoxins, the damaged enterocytes will shed into the midgut lumen, and then it triggers a JNK MAPK pathway and then transfer to the intestinal stem cells, and the stem cells became proliferation and also differentiation, and to finish the midgut regeneration. So we published last year, so I will not talk about that further. So actually, so this response is happened in the late state after cryotoxin treatment. So we found this around 48 hours post-infection. So after that, I would ask, so what happened in the beginning? So actually, there is another publication. I think this is, for me, from my knowledge, I think this is the only solid publication talk about autophagy. So in 2015, there is a publication talk about there is a transcriptional factor HLH30, so who regulates autophagy to defend to the cryotoxins, but it's in C. elegans. So I'm also asking questions, so whether autophagy function in a very important way to defense to the cryotoxin. So what is autophagy? So autophagy actually is an intracellular degradation system. It's very conserved almost in all the organisms. So it is a pathway that will transfer the damaged organelles or proteins or some invaded pathogens to the lysosome and to finish the degradation and to recycle the energy. So during this whole process, there are six steps. So first, the regulation of induction and isolation memory, and vesicle elongation and retrieval, and docking and fusion. So during this whole process, I would only to make a notice that ATG12, during this vesicle elongation, ATG12 and LC3, actually another name is ATG8, and also the sequencing 1. These three proteins, I will talk about that in the later part. So these three components are very, very important and core component in the autophagy. So this is adapter protein between the membrane, between the autophagy membrane and the substrate, which will be degraded in lysosome. So firstly, we found that when we treated the larvae, the name is rice stem borer, is insect pest on rice. So when we treat the larvae with cry9A protein with sublethal concentration, so we found some ATG8, the phosphatidyl ethanolamine ATG8 happened. So showing in with the confocal microscopy, we can observe the ATG8 puncta to show that autophagy happened. And also, we also, by using Western blot, we show that the PE ATG8 was observed in the midgut. So, and also we quantify the ATG puncta in the gut, and we found there is a significant difference between cry9 treated sample and the control. So also we're trying to observe the autophagosums in the midgut by using TEM microscopy, and we can find the double bilayer membrane were found in the midgut. So from those results, we can conclude that the autophagy is induced in the midgut after Cry9 protein treatment. So after that, we treat the larvae with the autophagy inhibitor after the Cry9 treatment, and we found after the treatments of inhibitors, the mortality increased, showing that autophagy is function as a defined response against Cry9. So after that, we analyzed we would like to try to find out which genes are the key genes during the response to Cry9 protein. So we performed RNA-seq RNA sequencing after Cry9 protein treatment. So we found two genes, as I mentioned above, the ATG12 and the sequenced 1. These two genes are upregulated. So and then we confirm the result by RT-qPCR, and we found these two genes are upregulated. So after that, we analyzed their function during the Cry9 protein toxicity. So we performed the bioassay after knock down these two genes by using RNA interference. So after knock down these two genes, we found only these genes, SQSTM1, showed increased mortality, but the ATG12, although slightly increased, but there is no significant difference. So from now, we can see the knockdown of SQSTM1 increased the mortality of larvae treated with the Cry9 protein. So after that, we analyzed the expression of SQSTM1 genes. So firstly, we analyzed the larvae, the expression of these genes in the larvae after different times of post-infection of Cry9 protein. We found in the beginning time, after 6 hours treatment of Cry9 protein, the expression of these genes upregulated. And we also would like to see whether these genes only function in the gut, and we analyzed the expression in different tissues. We found only in gut it has upregulation. So after that, we asked whether this response is only in the rat stem borer and response to Cry9 protein. So we analyzed it in for armyworm against the Cry1F protein, and we found after the knockdown of these genes, there is nothing happened. The mortality did not change, so suggesting that this gene may not function in for armyworm against Cry1F protein. So why? We analyzed the sequences of all the XQSTM1 protein across all the Lepidopteran insect pests, and we found that in the rat stem borer, this is the Latin name of rat stem borer, there is a domain showed here, but like in for armyworm, this domain was not. So perhaps this domain called the PHA superfamily, so maybe function as a very important role. So after that, we concluded that SQSTM1 but not ATG12 regulates defense action to Cry9 protein in rat stem borer. But there is something I forgot to say, because I mentioned the knockdown of ATG12 did not have any effect, but ATG12 in the autophagy pathway is very important. So we say maybe the SQSTM1 regulates the autophagy through a non-canonical autophagy pathway, or maybe it regulates some other pathway besides autophagy to defend the toxins. And also, we have another conclusion is autophagy plays an important role in responding cryoproteins, but not conserved in all the larvae. Okay. So after actually, it's at the same time. Besides the responding to the cryotoxins, I'm also very curious about what happened during the insecticidal action. What happened with the gut bacteria? Because there is a long-standing debate about whether the host insect gut microbiota is involved in the Bt insecticidal activity. So there is a paper in 2006, and they said gut microbiota is very important, but finally, Professor Ben Raymond and Neil Crickmore, they said, no, gut microbiota is not important. So the main thing about this is because the larvae was treated with antibiotic, but Ben Raymond, they think antibiotic also have effect on the spores of Bt. So it also affects the Bt bacteria. So that's why they have a debate. So actually, in recent years, very new papers, they are also working with only Bt toxin, without bacteria, without spores. And also the conclusions also are different, such as this part is that they found that if the gut microbiota was eliminated, the survival was going down after treatment of Cry1A proteins. But from other papers, they said they found different results after they eliminate the gut microbiota. The survival even increased after treatment of Cry1A. So they also found different results, except the effect of antibiotic to the spores. So after reading those papers, I found that Enterococcus is function like a lethal microbiota, but not 100%, but most of them. But Enterobacter function as a survival microbiota. It seems like that, but I don't know. I didn't know when I started our research. So actually, I started this work since 2020 about that. So we also did we performed the research with only toxin, without spores of Bt. So we also treat the larvae with antibiotic, and then we examine whether the gut microbiota has already been eliminated. So after the treatment of antibiotic, we can find nothing in the gut of the microbiota. So after the enzymtic strain of larvae was treated, so we performed this assay. So the control larvae treated with Cry1F is like this. So and also if we treat the control of okay, maybe we can talk about this. So if we treat the larvae with antibiotic and then treat the Cry1F, we can also find that the survival decreased, but it's later than the one compared to the control. It's like toxin can kill the larvae without gut microbiota, but later. So, and also we treat the enzymtic larvae by the addition of spores, and also the survival can recover. So it's like the microbiota and the spores plays like a similar function during the insecticidal action. But there is one thing is we need to mention, we need to talk about that. So when we treat the control larvae, it's non-arsenic, treat the Cry1F and with the spore, it did not show higher mortality compared to the only treatment with Cry1F. So it's like when the spores and the gut microbiota are all in the gut lumen, it didn't show any synergistic effect. So after that, we would like to further analyze which gut microbiota are functioning during this process. So we, by using 16S RNA qPCR, we found that after the treatment of Cry1F protein, after 48 hours since 96 hours, the gut microbiota are significantly increased. And also this is in the gut and also in the hemocoel. It's a little late than that in gut. So after the 72 hours, the microbiota, the bacteria in the hemocoel increased in the hemocoel. So after that, we also performed the 16S RNA sequencing to see which bacteria. So and we found that after we analyzed the alpha diversity, we found after the treatment of Cry1F, the diversity of the bacteria decreased. So it means that the diversity of bacteria was decreased in the gut of armyworm treated with Cry1F, but the abundance and the quantity of the Enterococcus were increased significantly because from this one we can see the Enterococcus and also the Enterococcus ASV number of Enterococcus are increased. So after that, we tried to isolate the bacteria in the gut, and we isolated 4 main gut bacteria, 2 Enterococcus and 1 Lysinium bacillus and 1 Streptococcus. And we, by addition of these 4 strains to the Cry1F treated larvae, and we found only this one, 6G10 strain, showed increased mortality after the addition. But the other 3 strains only, even also another Enterococcus, as E07 showed nothing, showed no different effect. So we sent these 2 strains for the genomic sequencing, and we found that this one who showed no effect is Enterococcus gallinarum. But the one we found who increased the mortality is a new Enterococcus species. There is no nomination till now. So from all the results we have is that we would like to conclude that different gut bacteria showed various effects on insecticidal activity, but it did show some effect, but not all of them show the effect. So we need to distinguish the different. Okay, so after these 2 parts of work, I would like to, from now on, I would like to give a perspective based on our work. So we think the regeneration of the mid-gut cells is very important in the defending responses. But autophagy, it happens in some larvae to some proteins, but not that conserved, and maybe it's not that important during the defense mechanisms. And also I found that gut bacteria and also the spores are also important during the insecticidal process. It will accelerate the insecticidal activity. But without them, toxins can also kill the insect, but later. Okay, so, but my perspective here is now from my work, and also I know Alejandra is also working with vesicle trafficking exosomes in the medical lumen. But from my work, we ignore that whether this response is only to the toxins or to the gut bacteria, we don't know. So we need to notice that. So because when we treat the larvae with cryotoxins, the gut microbiota is also in the gut lumen. So maybe in the future we will notice for this part. Okay, so finally, thanks to Alejandra and Mario for your organization, and also thanks to Professor Zhang Jie from our lab and Professor Alejandra Blava and Mario SOBERON for your long-term support to my study, because actually most of my publications were finished with all of your three guys together under the help of Alejandra and Mario. And also thanks to our colleagues in China and also the support from the foundations in China. Okay, thank you all.
Okay, are there any questions?
I have one. In this bacteria that increase the toxicity, the one that you discovered, the new one, can you find some virulence factors, or are you planning to detect? No? Maybe that bacteria is secreting other components that help to kill the larvae. And so maybe if you look in the differences between the two bacteria that you have sequenced already, maybe you can identify some virulence factors that could be very important for the toxicity.
Yeah,
no?
We are doing that.
You are doing that, yes. Mm-hmm.
Another question? I have a question. Because microbiota can change if you change diet. I wonder whether you feed the larvae with plants or with a synthetic diet, you will have different microbiota and different effects of the toxin.
Yeah. Yes, it's true. The different food they eat, the gut microbiota will totally change. Yeah, but all of the experiments we perform is with artificial diet. We would like to do that. And also we are planning to collect some strains of larvae from the field to see whether, because the larvae from different places, the gut microbiota are totally different. So maybe also they can show some difference. And also maybe we are planning to do that, to feed the larvae with artificial diet and to feed the corn leaves to see whether they are different. Yeah. Yeah. Thank you.
So now it's the turn of Isabel Gomez, no? Mm-hmm. She's the last one, yes.
Okay, thank you very much. I want to thank to all the speakers because we give you a very excited morning. I learned a lot of another bacteria because I work with BT, so it was very, very interesting. Thank you very much for visiting us. I hope you enjoy your studying here, okay? So let me go back to Bacillus thuringiensis, and my talk will be about exploring the role of the C-terminal region of cryprotoxins as determinant of specificity in the mechanisms of toxicity. So most of you know the cryproteins are produced as protoxins with two formats. One is long and is 130 kilodaltons, or the short protoxins that are around 60 kilodaltons. So an important thing here is why are long and why are short, no? But when the people start to study BT and the mode of action, we describe, and the most classical model accept that the, let me take this one. El de arriba. Ahí está. The classical model says that all the proteins must to be activated, and the toxin lose one part of the amino and most of the C-terminal region to be toxic. So in this model, as Sabino says, and another of our speakers says, that the crystals of the proteins must to be ingested by the susceptible larvae, and then these crystals are solubilized in the alkaline midgut juice, and after this, some proteases could this part of the protoxin, and the toxin, activated toxin, that is here showing just domain 1, 2, and 3, can bind to the membrane. Several proteins are described right now as receptors, as Sabino says, alkalinophosphate, aminopeptidase, or cadherin. These proteins work together, and the mechanism, as you see, is very complex. We described around 2010 with Sabino and Ivan as PhD students that ALP and RPN are the first receptors that interact with the toxin. This interaction helps to the toxin to concentrate in the membrane, and after this, the toxin can bind to cadherin in order to promote the oligomerization of the toxin, and finally the insertion helped by the ABC transporters to finally cause the death of the insect. So here, when I was working as PhD student, I was working with the interaction with cadherin, and we described the importance of this for the oligomerization of the toxin. But at the end of my PhD, I was asking me why this part of the protein is losing during activation. Why is not important? It's a big part of the protein, almost the half of the protoxins. So what happened with this? Is just losing in the midgut? Nothing happened? I was not sure about that. But in 2014, a group in United States described the three-dimensional structure of the Cry1AC protoxin. And when I saw the picture, I was very excited because I saw that all the C-terminal region is very structured. And one interesting thing is domain 5 and domain 7 are very similar to domain 2 in the activated toxin. As most of we know, the domain 2 and domain 3 are very important for specificity and the binding to the receptor in the midgut. So when I saw this, I was thinking, probably these domains are important too for the binding in the membrane. So we perform an experiment, a very simple experiment. We take the protoxin and activate the toxin, and we measure the binding with brush border membrane vesicle of Manduca sexta. And we observed that both forms of the protein can bind to the brush border membranes. So that means protoxin doesn't need to activate it to bind to the membrane. So that was the first observation. And after this, we compare the toxicity of the protoxin and activated toxin against Manduca sexta larvae, and we observed that the protoxin displays higher activity against Manduca sexta than activated toxin, almost 5 times. So that probably means that protoxin has an extra region that is important for toxicity that is losing during activation. So in this same way, in 2007, Tabachnik group described that Cry1AC protoxin can bind to cadherin in Pectinopora gossypiella. He used a recombinant cadherin expressed in E. coli, expressed the complete protein and several fragments, and he tested the binding using protoxin or activated toxin, and he observed similar that protoxin can bind and also the activated toxins. So we repeat this experiment using cadherin from Manduca sexta. We cloned several regions of this receptor, and we observed that activated toxin and protoxin can bind to this receptor with very similar affinity. No? So that means that protoxin doesn't need to be activated to bind to, at least binds to this receptor. So then we continue with the characterization of this interaction, and we observed that activated toxin can oligomerize after interaction with cadherin. So if we incubated this activated toxin with cadherin, we can get this oligomer that is around 150 kilodaltons and is very sensitive to temperature. We incubated at 25, 50 degrees, and 100, and at 100 we observed the destruction of this oligomer. But if we repeat this experiment using the protoxin, we see that we can obtain the oligomer just with cadherin. So after adding trypsin or midgut jukes that activate the toxin, we can get some oligomer that is more higher than from the activated toxin. This oligomer measures around 240 kilodaltons, and this oligomer is less sensitive to temperature. So we can even heat at 100 degrees and we still have some protein. So that means that the oligomer that is formed from protoxin and activated toxin is different. So we tested the in vivo activity of these proteins, and oligomer is more active than activated toxin. So that means the protoxin has an extra region that is important for oligomer formation and probably is involved in toxicity. So we propose that we have two pathways that for toxicity that working together. One is from protoxin that can bind directly to cadherin in order to promote the oligomer, and the second is working with activated toxin that can bind to the APN or APL, and after this, the toxin can interact with cadherin. So if this is true, in 2015, working with a Tabachny group, we proposed a dual model of action of CRY1A protoxins. In this model, protoxin and activated toxin bind to midgut receptors in order to provoke toxicity in sensitive insects. So we show here that protoxin and activated toxin has two different ways in order to insert into the membrane. And if this is true, if these pathways are working as independent mechanisms, probably insects that become resistant to activated toxin will be still susceptible to the protoxins. So Tabachny made a very nice experiment testing 7 resistant strains and showed that CRY1AB and CRY1AC protoxins were more potent than their corresponding activated toxins. So this experiment was crucial for our hypothesis because insects resistant to activated toxins are still susceptible to the protoxins. So that means in the C-terminal region are something that is working for toxicity. So I started a project with Arlene Peña. She made a PhD in our group, and I proposed the idea to test the role of the C-terminal region. So Arlene made the cloning of the C-terminal region of CRY1AB. So the first question was, the C-terminal region is stable in solution, because if this region is moved after activation, probably is very susceptible to degradation. But the first interesting result is that the C-terminal is very stable. Here you can see the expression in E. coli and was expressed very successful after expression. So we have the C-terminal region complete, and we can test the binding of this region against Bruce border membrane vesicles of Manduca sexta. We compare the binding between protoxin, toxin, and C-terminal. We used two kinds of antibodies, polyclonal antibody. One polyclonal antibody that was produced against toxin, activated toxin, and another antibody that was raised using C-terminal region. So you can see here that the protoxin and toxin are detected with antibody against activated toxin, but we can see the binding of C-terminal region because this antibody doesn't recognize this fragment. But when we use anti-C-terminal antibody, we can see the binding of protoxin and C-terminal, but not the activated toxins. No? So it's a very nice experiment showing that the amino and the C-terminal region can interact with the Bruce border membrane vesicles. So the next question was if C-terminal region can interact with the classical receptors in Lepidopteran, that is cadherin, alkalinophosphatase, and aminopeptidase. These 3 proteins were cloned in our lab, and we can express it in E. coli, because imagine if we can try to purify this from the larvae, we have enough proteins, but as recombinant we can have enough, and with these purified proteins we perform an ELISA assay and SPR assay to measure the binding with C-terminal region. Interesting thing here is C-terminal region can interact with APN and ALP, but not with cadherin. No? And this result was confirmed by SPR. We don't see any binding with cadherin, and the affinity with APN and ARP is around 3 nanomolar. It's higher affinity of this region with these receptors. No? So that means that it's important for binding. And a very nice experiment that we performed with Arlene is an immunoprecipitation with antibodies against receptors. So we have the recombinant proteins, we perform antibodies using some rabbits, and then we incubated protoxin, activated toxin, or C-terminal region with Bruce border membrane vesicle from Manduca sexta. After that, we run all the samples in a gel and transfer, and we used our antibodies to detect which receptor is with this, with every mode of the toxin. Protoxin was detected using anticadherin and toxin, and not with C-terminal region, as we show with recombinant protein. And APN was detected with the 3 form of the toxin, and ALP just with the C-terminal region. So again, we confirm that C-terminal region is involved in binding with aminopeptidase and alkalinophosphatase. No? So finally, we take some mutants that we have in the laboratory. I will show just one, that is a mutant located in domain 2 that is affected in toxicity. So is in domain 2 of activated toxin is affected in toxicity, and we tested the binding using our recombinant receptors, here is cadherin, ALP, and APN, using the form protoxin or activated toxin. No? So we can see here that cadherin doesn't bind as protoxin. No? APN and ALP can bind to the membrane, no? As protoxin or activated toxin. The mutant is affected in binding to the aminopeptidase and alkalinophosphatase, but is not affected in binding to cadherin. That means that activated toxin, if lose the C-terminal region, doesn't bind to ALP or APN. So with this, we conclude that the C-terminal region is important for binding, at least in Manduca sexta, with aminopeptidase and alkalinophosphatase. So probably the C-terminal region is giving an extra binding sites for the toxins, and if we lose these binding regions in resistant insects, has an important effect. So when we finished this part of the project, Arlene gets the PhD, she was very happy, and in the lab we started with another question. Is the same effect in another long protoxin? So, or if this is working just for Cry1A family. So we take another example. So we take Cry4BA, that is another long toxin, 130 kilodaltons, and we use another model that we have in the lab, that is Aedes aegypti. So also we have 2 clones, one for ALP, alkalinophosphatase, and a clone for aminopeptidase. So we express these proteins in E. coli, we purify it, and we cloned the C-terminal region of Cry4B. And when we tested in ELISA experiment the binding, we can see that the C-terminal can bind to both proteins. So very similar to what happened with Cry1A. Right now, we are working with this interaction just to show you if the model is very similar. No? Here you can see the binding of Cry4B protoxins is 1 nanomolar with alkalinophosphatase, and Cry4B activated, and Cry4B C-terminal region has very similar affinity for this receptor in Aedes aegypti. So right now we are running some experiments just to see if activated and C-terminal regions binds to the similar part or alkalinophosphatase. So you can see here an heterologous competition showing the binding of protoxin. Here you can see the protoxin binding to alkalinophosphatase. And if we compete the interaction using 100 excess of protoxin, we can see here how the binding is lowest. The same happened with activated toxin or the C-terminal region toxin. No? So as Aminos mentioned, probably this binding is not important for toxicity because he has the knockout, so we are not sure if this is important. But I believe that the C-terminal region is working for attach the protoxin into the membrane, and probably this help to the toxin to go down in the mechanism of toxicity. And I believe too that these receptors are working together. It's not just one. So the mode of action requires more than one receptor for toxicity. So it's the first step to show this one. No? So finally, as conclusions, we know now the C-terminal region of Cry1AB protoxin is important for the interaction with alkalinophosphatase and aminopeptidase in the membrane. No? So here we are showing that. So the protoxin can bind to this receptor using the C-terminal region, and this promotes the binding to cadherin and then the oligomerization and finally the insertion. So right now we are working narrow the C-terminal domain involved in binding receptors. So we are running an extensive mutagenesis in domain 5 and 7, trying to find mutants that are affected in toxicity, in binding and toxicity, and we are trying to conclude the results about the role of the C-terminal region in another cry toxins. And I think that that's all for me. And if you have questions, I will be happy to answer. You are tired, no? We are hungry, right?
Yeah, are there any questions?
Thank you, Isabel. Very interesting. So if you reduce, or if the C-terminal region is not present, then there is the decrease in size is very important, as you said. So in the tridimensional structure, have you seen if the C-terminal region is protecting in any way the rest of the protein from proteolysis, for example? And when viral entry is modeled, something that is very important is Brownian diffusion. So I don't know if, because this much larger protein would have a much slower diffusion compared to this smallest one. So that could also affect in real life if this protein is going to bind to receptors. I don't know if somebody has studied this.
I think that is a very complicated question because we are not sure what happened when the insect eats the crystal, because the protoxin is embedded in a crystal. So we believe that when the crystal is solubilized, so the protoxin probably is open, and structure open, not unstructured, and probably that go inside to the midgut. But we don't have an experiment that proved that. No? So we are working, trying to imagine how we can prove that the protoxin go in entire form to the midgut, no? And prove that the structure is conserved in the membrane. No, so it's not coated before or after. No, I'm not sure. But with this, I feel that is important. No, it's an important role. That's why I am running the extensive mutagenesis in that region. If I have mutant that is affected in toxicity, so probably I can prove that. No.
Thanks for your presentation. Actually, I have a, how to say, a hypothesis, or actually Alejandra and Mario should know our progress in our lab. We found that the C-terminal can bind sugar. You know?
Yeah, probably.
Yeah, because from your results, ALP and APN are GPI anchored receptors.
Yes.
So I think because they have a glycan.
The structure is very similar.
Yeah,
to
and
the
also
glycan.
C-terminal has a sugar carbohydrate binding motif.
Yes.
So I think maybe the carbohydrate or sugar or say glycan may be function during this whole process.
Probably.
Yeah.
Probably.
Sorry, I will give you. Just, well, she found binding to the recombinant proteins, which has no sugars. So it binds protein-protein interactions because she produced the receptors in E. coli, which are not without sugar, and she has very nice binding.
Okay. Nice work. It changed my mind to recognize the C-terminal, what is the function. But in my mind, the activation, it's very fast when the crystal is so localized.
Yes.
Maybe just stabilize this activity.
Yeah, but in that part, we have another big hole, because probably the activation is very fast, but the protease is in the midwood juice or is attached to the membrane. So where is the activation process?
So did you compare the activation? Someone is maybe very strong and others is very weak. So for those who has a very strong activation, maybe the C-terminal maybe no function. Maybe
I'm not sure. That is a very interesting question, but I'm not sure how to prove it.
Oh, okay. Because in my mind, actually, when I studied in a master's degree, it's 30 years ago, I have compared with that CRY1AA to protoxin and active toxin to against the David-back mouse.
Okay.
But I found that S50, it's nearly the same in my mind. I never seen this 30 years ago.
Yes, I'm not sure.
It's good. Okay, thank you.
Benito.
Okay, thank you for your conference. What could happen if you mix C-terminal region with the protoxin and toxin? And another question is, if you incubated first with C-terminal and after feed with the
C-terminal?
No, first C-terminal and after feed the larvae with the protoxin.
Okay, with Atlan we run an experiment testing the toxicity of the C-terminal region, but the C-terminal region is not toxic. It's not toxic alone. That could be. But we perform the bioassay using the C-terminal. It's not toxic. And when we mix the C-terminal with amino, with the activated toxin, and the toxicity was similar. So we didn't see any increase of toxicity. But we don't try with protoxin, so I'm not sure. No, probably the toxicity can go down, but I don't know.
Okay, muchas gracias, Isabel.
Gracias. Thank you.
So we finished the workshop. I congratulate all the speakers. We have a great session. We thank our colleagues from China. They have a very high jet lag, so it was a great effort. Yes, actually, Laura will join us for the picture too. No? So probably we can go.